Yeah, excellent. Arushi, can you just add to dispatches? Yeah, I'll just prepare the slides. Great. We are there. We are on YouTube.
Hi, Jyotsna.
Hi. Sorry, people. I got a bit delayed. Hi, Karishma.
Hi,
Hi,
Jyotsna. Good to see you. Good to see you.
yeah, hi. Hi, Prasad. We've not met before,
Yeah,
but
yeah, nice.
nice to meet you. Yeah, and
Pleasure.
hello to all the participants. Sorry, people. Hi, Karishma. Hi, Jyotsna.
Yes, we'll just get started. Uh, the
Hi, Raj.
slides suddenly don't seem to be opening, but yeah, let me just figure out what I can do quickly. Arushi, could you download the slides at your end? Day 5, session 2. For some reason, they are not opening at my end.
Yeah, just
We,
give me a
we,
second, I'll do it.
yes. Thank you. Thank you. Sorry for this small technical glitch. But in the meantime, while Arushi is handling that and Rutuja is looking at the meeting room, we will check if all our speakers are in. So I can see our expert mentors are in. This is not a roll call. I promise you, this is not an attendance, but this is just a check so the technical transitions become easier. Umanshi, I see you. You're there. Yes. Umanshi, you're there.
Yes, yes, ma'am.
Great. Akshaya, you're there. I can see you too. Vishwadeep. Vishwadeep
Yes.
is there. And Olivia. Olivia is there. I think she will be not here. Olivia should be joining in soon. Okay, she's not yet there. All right. So, welcome to the final session in PhD Cafe. This is Developmental Biology. Thank you so much for your time. It's been an action-packed week. We started on Monday with back-to-back scientific sessions and open networking sessions through the afternoon, in the afternoon hour. And we've had with this session, we'll have 50 speakers who have spoken. Thank you so much, Arushi. Thanks. Thanks a ton. So trust my computer to know that it is the last session of the series. And it decides to not open PowerPoint. We'll figure that out later. Anyway, this is what is lined up for today afternoon. We have 4 speakers and a range of very interesting talks, some really exciting talks. So welcome to all of you and all the best. So each speaker is given 20 minutes. We can move to the next slide, Arushi. So each speaker will be given 20 minutes for their talk. We have strictly told them 14 slides. Most are adhering to that. And I would also say that speakers have done an excellent job of sticking to time. So after 20 minutes, which my colleague Manjula will keep time, we have 10 minutes for open Q&A. That will be primarily from the expert mentors. So all of you, Raj, Prasad, Jyotsna, and Surendra. So after your open Q&A, suggestions, feedback, considerations, comments, welcome. We will then see if we have time for audience Q&A. But otherwise, audience, please keep typing your questions in the chat window because the speakers can always attend to those questions after their talk, right? So keep that active. And thank you all. We know you have a lot going on. Thank you for making the time to mentor PhD researchers. The idea behind this was to apply for a company of biologists grant and to fill in this space in Indian life sciences where PhD researchers don't get enough opportunities to give talks. And so to give them a sort of pre-synopsis or pre-defense, depending on the stage in their PhD career they are, experience, and to do it in a safe, supportive space so they can make mid-course or late-course correction that can help them in their PhD. That was the whole idea. So we couldn't have done it without our mentors and speakers who came on board. Thank you all. Okay, moving to the next slide. We have an open Q&A session, as we said, through the chat window. We also have a giveaway, and that is a very exciting e-compendium from India Bioscience, which was published by my colleague, Ankita Rathod. So that book deals with challenges in the life science ecosystem in India. And the audience member who keeps the chat window most active gets the chance to win this e-compendium. Today is the 10th and final giveaway. We've already had 9 giveaways done. So 9 people have won a physical copy of this e-compendium, and today is the 10th winner. What the audience needs to do to get this is keep the chat window active. All right. So with that, I think, Arushi, let's get started. So the first speaker is Umanshi. Umanshi, you might want to start slide sharing. And yeah, great.
Yeah, sure. I'll share my slides.
Yeah. Perfect. Yes, we can see it, Umanshi. You can start, and 20 minutes starts now.
Okay. So a very good afternoon to everyone. And I would like to thank the organizers for giving me this wonderful opportunity today to talk about my doctoral work here. And today I'm going to talk about the role of this G1-S-phase cyclin D, and how it cross-talks with the nutrient-sensing insulin IGF-1-like signaling pathway to influence the fidelity of reproductive tissues, as well as to regulate oogenesis. So in females, we know that the reproductive tissue is one of the first organs to show the signs of aging. And it is manifested as abnormality in oocytes, reduction in fertility, chromosomal abnormalities, birth defects, as well as tumors of the reproductive tissue. There are various factors which can actually accelerate reproductive aging, such as poor nutrition, stress, hormonal imbalance, as well as exposure to different toxins. To study the underlying mechanisms of reproductive aging, in our lab, we use the wonder worms Cinorhabditis elegans, which is simple soil-dwelling nematodes, and they can help us solve the mystery of reproductive aging. So shown here is one of the gonadal arms of the adult C. elegans. And in this schematic, you can see that the germ cells, the mitotic zone carries the germline stem cells which divide. Then as the germ cells enter from the mitosis to meiosis, there's a transition zone which has a peculiar crescent shape due to the homologous chromosome pairing. And the germ cells enter meiosis in the pectine zone where homologous synapses take place. Then there is a turn region during which 50% of the germ cells are culled or optosized to maintain the quality of the germline. Since most of the worms exist in a hermaphrodite population, meaning that they carry both the sperms and the oocytes, and they are self-fertilizing. So there is a production of oocytes, as well as there are sperms which are stored in the spermatheca. And upon dissection or staining of these gonads with the nuclear stain like DAPI, we can see a beautiful morphology of these germ cells, and we can see a peculiar patterning such that there's a mitotic zone at the distal end, the crescent-shaped germ cells identified in the transition zone, and a spaghetti-like structure of the germ cells, which are in the pectine zone, followed by bivalent chromosome structure carrying oocytes and the sperm stored in the spermatheca. So why C. elegans? So there are different hallmarks of reproductive aging, which are actually conserved from worms to humans, such as the germline stem cells reside in a niche in both. Apoptosis is used as a maintenance system for oocyte quality maintenance. The oocytes are rested in meiotic prophase 1, and the oocyte quality declines with age in both humans as well as in worms. So giving a brief about the C. elegans cycle, the adult reproductive C. elegans lays eggs, which goes through different larval stages L1, L2, L3, and L4 to give rise to the reproductive adult in a matter of 3 days. And the germline basically develops along with the somatic gonad such that there are 2 somatic gonad precursor cell and 2 germ cell precursor cell during the L1 stage. And as the C. elegans morphoses to the L1, L2, L3, L4 stages, the germ cells divide and enter meiosis in the proximal region, as well as the somatic gonad cells divide and nourish the growing germ cells and giving cues for the division or the differentiation. During the late L4 stage, the spermatogenesis takes place and gives rise to a fixed number of sperms. At that late L4 stage, there is a switch that has to take place, which determines that the next number of germ cells which are going to be having the oogenic fate. And then there is a continuous production of oocytes. The fertilized eggs are stored in the uterus. So there are 5 important somatic gonad components, of which I'm going to talk about the sheath cells, which basically enwrap the germ cells, and the uterus, which are basically storehouse of the fertilized cells in my talk today. So as I mentioned, that many of the hallmarks of reproductive aging are actually conserved in worms to humans. And one of the pathways which is shown to influence the reproductive aging is the insulin signaling pathway. In worms, the insulin signaling pathway begins with the binding of the insulin-like peptides to the DAF2, which is the insulin receptor. And there is a series of phosphorylation that takes place via different kinases, ultimately leading to the inhibitory phosphorylation of the FOXO transcription factor, which is DAF16 in the case of worms. So in wild-type conditions, when the insulin signaling is normal, the DAF16 is cytoplasmic. But in the DAF2 mutant that we have used in our study, the insulin signaling is lowered, meaning that the FOXO is not having this inhibitory phosphorylation and it is nuclear translocated, giving rise to the multiple benefits like longevity, stress resistance, and better germline quality. So throughout my talk, please remember that the DAF2 mutant worms will be having a lowered insulin signaling, and in this case, the FOXO will be activated, unlike the wild-type case. So as I told that the worms have similar reproductive aging hallmarks, shown here is the DIC image of the wild-type gonad. And because of the transparent nature of the worms, we can easily look at the morphology of the oocytes. So at day 1, the oocytes of the wild type look properly stacked without any cavities or without any misshaped oocytes. But as the worms age, for example, at day 8, we can see that the oocytes become severely poor oocyte quality, they become misshapen, and there's a lot of disorganization in the gonad. Whereas the DAF2 mutant worms, which have lowered insulin signaling, are better able to maintain the quality of the oocytes morphologically as well as functionally, because with age, the wild type start laying a lot of dead eggs or eggs that do not hatch, but the DAF2 worms still have pretty good embryonic hatching. So this shows that FOXO is important to maintain the germ-like quality. In a previous study from our lab, we asked whether what interactors of which kinases would interact with the insulin signaling pathway. And serendipitously from our screening, we discovered that CDK12, which is the C-terminal domain kinase, which phosphorylates the RNA pol to CTD, and it is known to be involved in the DNA damage response, and it is known to be involved in the transcription of the genes important for DDR. Interestingly, what we found is that the DAF2 mutant worms, when knocked down with CDK12, they basically lead to germline arrest. In this study, we show that the CDK12 knockdown leads to instability of the genome, which is sensed by the activated DAF16. And to prevent the production of poor oocyte quality or the damaged oocyte quality, it leads to the arrest in the oogenesis. So we became interested that if the kinases can crosstalk with the insulin signaling pathway to regulate the reproductive outcomes, what about the cyclins? Or do the cyclins also crosstalk with the insulin signaling pathway to regulate the reproductive outcomes? For which we did a small RNA screen and knocked down the cyclins in the wild-type background, the DAF2 mutant background, as well as the DAF2 DAF16 double mutants, which do not have the FOXO transcription factor in the DAF2 mutants. Interestingly, we found that the G1S cyclin D, when knocked down, leads to stimulus sterility only in the DAF2, and this is DAF16 dependent, such that the DAF2 DAF16 double mutants are fertile. So, in the project, we addressed two main questions, which I'll be covering in the talk today. So first, we try to address that what is the role of cyclin D in the germline quality of wild-type worms where the insulin signaling is normal. And what role does cyclin D knockdown take place that leads to the germline sterility in the low insulin signaling when the FOXO is activated. So going on to the first question and determining the role of cyclin D in wild-type worms, as we know that cyclin D, along with its binding partner CDK4, it is important for the phosphorylation of the retinoblastoma protein and in the transition of the G1 to S phase. So we try to ascertain if the germ cell number is affected or not in the wild-type worms. So shown here is, just to remind you, is the dissected image, and there is a mitotic zone at the distal end, calling pectine zone, the turn region, and the oocytes. So when we see the knockdown of cyclin D in the wild-type worms, although the worms are not sterile and have both the sperm and the oocytes, upon quantification of the germ cells in each zone, interestingly, what we observed is that the mitotic zone germ cell number did not change when we knocked down cyclin D. However, interestingly, the pectine germ cell number decreased upon this knockdown, which was very fascinating, and it hinted towards maybe a non-cell cycle role of DAF16 in this case. As we know that following the pectine zone, there is a turn region where the apoptosis takes place. And we asked whether an increase in apoptosis is the reason due to which we are seeing this reduction in pectine germ cell number. For that, we looked at the apoptosis using a transgenic strain carrying SED1-GFP. SED1 is a transmembrane protein on the sheath cells which engulf the dying cells. And it is the apoptotic corpses are visible as these green dark foci. So we see that in cyclin D knockdown, we do see an increase in the apoptosis. And since we know from previous studies that apoptosis is required to maintain the quality of the oocytes, and it is basically a culling mechanism to remove the damaged or defective germ cells. So we ask that, is cyclin D knockdown compromising the oocyte quality in the wild type? For that, we assess the oocyte quality first morphologically, and upon imaging, we divided and scored the gonads as normal, mild, or severe based on the presence of cavities or the disorganization of the gonad. In the cyclin D knockdown worms, we do see a lot of misshaped oocytes and disorganization of gonad, which is quantified here. And we see a lot of severe quality oocytes present when the cyclin D is knocked down. Interestingly, even when we are mating these hermaphrodites with normal sperm where cyclin D is not knocked down, still we see that the oocyte quality is not improved and the disorganization and misshaped oocytes are still there, meaning that the sperm is not affecting the oocyte quality here, but the oocyte quality is inherently poor. We also checked the functionality, that are these oocytes only looking poor or are they really poor in oocyte quality? For that, we looked at the live progeny per worm and observed that in both selfed as well as mated worms, we see that the cyclin D knockdown is not able to rescue to the wild type control level. Also, the egg hatchability decreased upon cyclin D knockdown. Also, with age, we observe that a lot of endomitotically dividing oocytes are accumulated in the uterus. And in the cyclin D knockdown, we observe that these endomitotic oocytes were highly upregulated when we knocked down the cyclin D in the wild type worms. So all these phenotypes hinted towards the role of cyclin D in maintenance of the oocyte quality under wild type condition or when the DAF16 is cytoplasmic. So since our previous results hinted towards a non-canonical role of cyclin D, we thought that in which tissue cyclin D might be important to maintain the oocyte quality. For this, we knocked down the cyclin D in different tissues, such as only in the germline or only in the soma, as well as in the whole body. Interestingly, only in the germline when we knocked down cyclin D, we do not see deterioration in the oocyte quality. However, soma-only cyclin D knockdown led to deterioration in the oocyte quality with high cavities and misshaped oocytes, similar to the level of the whole body knockdown, and which was interesting. So these lines we were validated with using different control experiments, which I have not shown here. But we have validated that these tissue-specific knockdown lines have only tissue-specific knockdown of cyclin D. Then further, one by one, we knocked down cyclin D in different somatic tissues to find out in which somatic tissue, like whether it is in the neuron, muscle, or intestine, where is cyclin D required to maintain the oocyte quality of the germline. interestingly, what we find is the uterine tissue only knockdown of cyclin D led to deterioration of the oocyte quality similar to the wild type whole body knockdown. Here I want to remind that uterus we believe just to be a storehouse of the eggs, but it seems like that it is not a just bystander for the oocyte storage, but it is playing an active role in the oocyte quality maintenance. And this we also see what this is also parallel to what we see in mammalian studies, where the uterine health, as we know from the recent studies nowadays, is impacting the oocyte health as well as oocyte implantation rates, the egg implantation rates. The same is quantified here, where we see that uterine tissue-specific cyclin D knockdown leads to deterioration of oocyte quality, as well as we also observed increase in the endomitotic oocytes in these worms. To summarize my wild-type data, we have observed that when the DAF16 activity is low or the DAF16 is cytoplasmic, the cyclin D levels, if they are maintained, it can maintain the healthy oocyte quality. However, if we are lowering the cyclin D only in the uterine tissue, the oocyte health deteriorates, as well as there is an increase in the endomitotic oocytes, which shows that cyclin D is important to maintain the oocyte health in the wild-type condition. And coming to the second objective, what is happening when the DAF16 is activated or when the worms have lower insulin signaling? As we know that low insulin signaling gives multiple health benefits such as longevity. So we decided to ask now that what is happening under the low insulin signaling. We observed that in the DAF2 mutant worms, whereas the control worms had the germ cells, the oocytes, as well as the sperms, the knockdown of cyclin D led to the abrogation of the formation of oocytes, leading to pectine arrest, and there were only sperm formation taking place. Upon quantification of these germ cells, we found again that the mitotic zone, interestingly, was not affected, but there was a stark difference in the pectine germ cell number. We again asked, since the sterility was DAF16 dependent, what happens in the DAF2 DAF16 double mutant where there is no FOXO? And we find, indeed, the FOXO removal in the DAF2 mutant worms brings back the oocytes and also the pectine germ cell number. And now we thought that since the FOXO is not there and we are getting the oogenesis rescue, what is the quality of these oocytes? Are they also poor oocyte quality similar to wild type? And indeed, that was the case, such that when we are knocking on cyclin D, in the DAF2 DAF16 double mutants, we see a lot of misshaped oocytes with a lot of cavities, which means that if you remove FOXO from the DAF2 mutants, it is no longer able to arrest the germline, and then it starts production of the poor oocyte quality, which again re-emphasizes the role of DAF16 as a guardian of germline and to protect the progeny fitness. We then asked where cyclin D is required in this tissue. Is it again cell non-autonomous, or cyclin D knockdown in the germline can lead to this germline arrest? Similar to the wild type, we found that cyclin D is not required in the germ cells to mediate this arrest, whereas if we knock down the cyclin D only in the uterus of these lower insulin signaling background, we are able to get a similar pectine arrest as we see in the whole body DAF2 cyclin D knockdown. So here we thought that activated DAF16 is able to sense maybe the uterine health and any perturbation. For example, by knocking on cyclin D is able, the DAF16 activated form is able to sense this perturbation and halt the oogenesis. So to better understand the underlying mechanism, we did a transcriptomics analysis of the DAF2 when cyclin D is knocked down. And we found, interestingly, many of the somatic gonad genes which are important for the somatic gonad development were indeed downregulated when cyclin D was knocked down. And many of these are important for example, sheath cell development or uterine development. We looked at few of these markers, such as LIMP7-GFP and FKH6-GFP. And we found that upon knockdown of cyclin D, the LIMP7-GFP expression was completely abrogated in these DAF2 worms. And upon removal of FOXO, we get back the expression back. So here, I just want to remind that sheath cells, there are 5 pair of sheath cells encircling the growing germ cells as well as the oocytes. And sheath cells are very important for various processes in the germline, such as the germ cell proliferation, apoptosis, sperm to oocyte fate switch, as well as the oocyte maturation and fertilization. So here, we again looked at whether the sheath cell structure or development is fine or not. For that, we stained these sheath cells with phalloidin staining, which marks the F actin. And we found that the sheath cell structure was abnormal when cyclin D was knocked down, but it was rescued back to normal in the DAF2-DAF16 double mutants. So this was very in contrast to the grand pro-longevity role of DAF16, which is known. And this is one of the few studies where we see that FOXO is actually helping, leading to germline arrest by deteriorating somatic gonadal tissues. So here we asked that, and this deterioration of somatic gonad could be the signal for the germline arrest. So next we asked that why is the germline arresting in the DAF2? So as we know that in the hermaphrodites, both sperm and oocytes take place. The late L4 stage is the stage where the sperm to oocyte switch has to take place. If there is no switch, then there is a mock phenotype where continuous production of sperm takes place, and we say that the germline has been masculinized. There are different players which play a role in the sperm to oogenesis fate switch. So to first, there could be two possibilities, that either the germline is masculinized or the germline sperm to oocyte fate switch is hampered. To look at the first possibility, we looked at the number of sperms in the DAF2 worms when cyclin D is knocked down. And upon quantifying the number of sperms, we found no difference, and there was no age-dependent increase in the cyclin D depleted worms, which means that there is no continuous production of sperm, although there is absence of oocytes, which leaves us to a second possibility that maybe the oocyte to sperm switch does not take place. And we found that many of these genes were actually downregulated in the DAF2 mutants when cyclin D is knocked down, and they were rescued back to the wild type level in the DAF2-DAF16 double mutant, which shows that maybe the sperm to oocyte switch is affected when we knock down the cyclin D in these DAF2 mutants. Also, interestingly, the uterine-specific knockdown of cyclin D was sufficient to lead to the downregulation of these genes important for the sperm to oocyte switch. To summarize my talk today, what I have shown is that in the wild-type condition, when DAF16 is cytoplasmic, the cyclin D in the uterus is important for maintaining the oocyte health. However, when the DAF16 activity is high, such as when the worms are calorie restricted or when there is low insulin signaling, we see a very counterintuitive role of DAF16, where the DAF16 activation senses the deficiencies in the uterine tissue and destroys the somatic gonad tissue such as sheath cell, which leads to a defect in the sperm to oocyte fate switch and halting of the oogenesis. So we talk about a cell non-autonomous role of cyclin D oocyte quality maintenance, as well as the role of FOXO in sensing the perturbations in the somatic gonad and further leading to somatic gonad distortion to orchestrate sterility in the DAF2 mutant worms. And probably this could be a mechanism to prevent the production of faulty progeny. I would like to thank you all. And I would like to thank my lab members, my supervisor, Dr. Namak. I would like to thank Gautam, Ayushi, and Debilina, Rosh, and Dr. Anish for their help in this project. Thank you, everyone. And I would like to thank all my lab members and the central facility. Thank you, everyone. So, yeah, I'm open to questions.
Well done, Umanshi. Great. I think you finished quite close to 20 minutes, so excellent timekeeping. Let's take questions from all our mentors. We have 10 minutes to answer these questions. So the more questions you want, try and keep your answers to the point. All right, let's go ahead. Raj, Jyotsna, Surendra, Prasad, go ahead, please.
Yeah. May I start, because I have some very general questions. Very nice presentation and very nice work. Okay.
Thank you, sir.
Congratulations.
Thank
So
you.
I was wondering if you have tried the overexpression of cyclin D to rescue some of the phenotypes.
Okay, so we have not tried the overexpression as of now because we have not used mutants. So the cyclin D mutants are lethal. So that's why we resorted to using the RNAi.
Yeah. But what I was thinking was whether there is any rescue that can be seen, because in the worm, it might be easier to. I'm not familiar with the system, so I'm just wondering whether it can be done.
Yes, sir,
Okay.
it can be done. Yeah, thank you, sir. But
And
we have
one
not
thing
tried
you
it.
mentioned in the beginning, that there is a fixed number of sperm. What do you mean by that?
Yes. So in C. elegans, the late L4 stage, so basically around 200 sperms are formed during late L4 stage. after which there is a sperm to oocyte switch that takes place. And there is
But
no more sperm formation.
but
So
if
sperms
there
are
is
formed
a
only during that time.
but if there is a long-lived male, what happens?
Okay, in males, there is continuous production of sperm, but in
Okay.
hermaphrodites, there is a fixed number of males. Yes. Sorry,
Okay.
sperms.
And there is a very general question, once again, may not be applicable to your model system. See, we know that there is longevity, but throughout the life cycle of many organisms, there is no reproduction. How is it in worm? Do they reproduce till they die?
Okay, no. They also have a fixed reproductive phase followed by a non-reproductive phase. So
Okay.
basically,
So there is one school of thought, and many people are suggesting that in animals like elephants or even human beings, the post-reproductive phase could be very useful to for parenting, for the group per se. Has anything like that has been considered in the worm? Do they depend on each other? Is there any communication between them?
Yes, sir. So there's a study from David Jem's lab where they show that yolk venting occurs in C. elegans. So basically, the mothers, even if they're not actively producing, they keep venting the intestinal yolk, which they would have used for the progeny. But since they are no more reproductive, so they vent this lipid-rich yolk, and it helps in the younger worms to survive or the progeny to survive. So
That's
there is yolk
fantastic.
venting in the worms.
I'm very excited about that because this is very rather more clearly shown in more complex animals like elephants. The senior most female in the group. She's the one who guides them to source of water and all that. It's not directly related to your work, but I thought that as a biologist it might be worth considering. Okay.
Okay.
Thank you. Thank you. I'm done, Karishma.
Excellent. Thank you, Surendra. Thank you. So Raj, Jyotsna, Prasad, please go ahead.
If I can ask. So, hi, Umanshi. Very nice talk and very interesting work.
Thank you.
Yeah. So rather than the biological sort of high-level questions that Suren just asked, I thought I would ask you the question about the molecule that you've been studying to uncover this sort of non-cell autonomous role for a mitotic regulator, right? I mean, or a cell cycle regulator like cyclin D. It's quite intriguing, right? And if I understood you correctly, because you've shown there's no effect on the frequency of mitoses, and the effect happens later, only in meiosis, you have this packaging-specific phenotype. What do you think might be the targets of cyclin D, which are mediating this, right? I know you've shown the interaction with the DAF16, DAF2 axis, but how do you think cyclin D is doing this work if it's not acting in its canonical role as a G1S transition mediator?
So, ma'am, like what I have not shown here is a few results where we see that although we think that it's not a canonical role of cyclin D in the germline, but it might still be a cell cycle dependent role in the somatic gonad, because only when we knock down cyclin D early during development do we see all these phenotypes. If we knock down cyclin D in adult, then we do not see acceleration of reproductive like aging phenotype, or we do not see the arrest in the germline tissue when it has been formed. So only in the development, if we perturb this cyclin D levels, do we see these effects, which also mean that maybe cyclin D is very crucial for the somatic gonad development. And because somatic gonad is important for maintaining oocyte health or maintaining like proper oogenesis, that could be an indirect effect.
So if that's true, and that it's not that you're not talking about a specific role for other targets of the cyclin D CDK complexes, and that it's working through its canonical functions. Do you think that modulating, affecting, knocking down other components of the same complex of the somatic tissue? And are such mutants available in C. elegans? I assume all mutants are available in C. elegans.
So we checked for CDK4, which is the canonical binding partner of cyclin D. So
Yes.
although we have not checked the tissue specificity, but due to the knockdown efficiency, we have to use a hypersensitive mutant here in the DAF2 background. And we do see that it causes similar pectine arrest like we see in cyclin D. But we have not checked the tissue specificity there. And also cyclin E and CDK2, which are further downstream of cyclin D in the G1 to S phase transition. Interestingly, we have seen that they act cell autonomously and they regulate more like germ cell number. So, and this could be because of the patterning of the C. elegans germline. So most of the germ cells in the mitotic zone, they are already in the G2 phase. And which could be that we do not see direct effects of cyclin D here. And that is why maybe we could study it and see its effect on meiosis instead, because we could see the proper germ cell number and we could differentiate between wild type and low insulin signaling condition. So
Right.
mutants are actually lethal for cyclin D and CDK4, and they arrest very well in larval stages. So that's why we have used RNAi.
Right. So the other issue that arises is, do you think that the signal that is going from the uterine cells, the somatic cells, to the germ cells, which is regulating their development and their progression, right, and their overall health, is it entirely through the insulin IGF signaling, or are there other signals that might actually be responsible for being connecting to cyclin D, that cyclin D might be affecting?
Other signaling one we have not checked actually. Like apart from the insulin signaling, we have not checked the other crosstalk. But we did check that whether the insulin signaling senses the uterine health in general. So for that we have used some knockdowns of some of the very important genes which are important for somatic gonad, and we see that only when the somatic gonad is perturbed at a very drastic level do we see this germline arrest and crosstalk with the insulin signaling. So that we have checked. But what cyclin D?
So I guess
crosstalking
just to interrupt you, I'm just trying to get a sense: is the insulin signaling sufficient to explain the entire signaling between the somatic cells and the gonadal cells, or do you need to invoke other signals?
Okay. So
quantitatively, when you look at the recovery, when you affect the signaling, the insulin signaling in the background of the cyclin D, is it a complete recovery, or is there still some room for improvement?
okay. So
Okay,
actually
Umanshi,
we have
maybe you
checked-
can think about it a little. It's a
Yeah.
good question. Maybe you can note it
Yeah.
down and think about it.
Right.
Yeah, sure, sure. We can then maybe, Josna, thanks. We'll just move to Prasad and Raj. Yeah,
Sure.
because our Q&A time is also a little tight. Prasad, Raj, any
Yeah.
suggestions? Umanshi,
Yeah,
do note these down.
Okay.
Yeah.
Yeah, Umanshi, great work and great talk.
Thank
So just have a quick question about the switch that you mentioned between sperm to oocyte, right? That switch is also regulated. That's what you are hypothesizing, right?
Yes.
So that switch has to be somehow regulated at epigenetic level,
Okay.
right?
We see, sir,
Because multiple
transcriptional
gene,
changes.
exactly, multiple transcription changes has to happen, right? For the switch to
Yes.
to take place, right?
Right.
So what do you think? How does your cyclin D is probably controlling that epigenetic regulation? So that is global. You are seeing multiple changes in multiple transcript, right? So how
Right.
that global change is like taking place? There has to be some link. Do we have any knowledge about it, or you're looking into that aspect?
Yes, sir. So as of now, what we see is that it is maybe a signal which is important from the uterine to the germline, because we see that when we knock down cyclin D only in the uterine of these DAF2 worms, still we are able to see the deregulation of the sperm 2 side switch and the arrest. So there has to be some signaling defect also, due to which we are not able to see the oogenesis. But as of now, we do not know what is the signal. What we are hypothesizing that it is again an indirect effect, and maybe this is a effect due to defect in the sheath cell structure and the sheath cell function. And since sheath cells are important for the sperm to oogenesis switch, so this could be a possibility that it is leading to the germline arrest due to the defect in the sheath cells.
Okay, in that case, do you think this systemic metabolic change that is maybe probably occurring in sheath cells, that could be driving this switch, something like that?
Yes, sir, that is what we are proposing.
Is there any genetic targets you can directly test out this hypothesis with? Like, do we know any genetic machinery involved in this switch you can directly test in your mutant condition? That could be one
So
way.
yes, since it's a very interactive kind of a network, so we do not know if rescuing just one would be sufficient. So till now we have not
Right,
tried
multiple.
rescuing each of these. Yes,
Okay.
maybe regulating, if we upregulate one of the master regulators, it could
Correct.
rescue also. We have not tried as of now.
Okay.
Thank you, sir.
Great. Yeah, best luck. Best luck. Yeah.
Yes, Raj, please, over to you.
So a very quick question about the cyclin D interactors for this non-cell autonomous role. You talked about CDK4. Did you look at CDK6 as well?
So actually in C. elegans, we have CDK4 as an ortholog for both.
Okay.
Yes.
Okay. Yeah. Do you have any kind of this non-canonical function? Do you have a clue of how it's working apart from maybe sheath cell dysfunction or anything like that?
Yes, sir, we do think that it is important for the somatic gonad development. And there has been previous studies where Judith Kimball's group, they have shown that in C. elegans, the early divisions of somatic gonad are actually dependent on cyclin D. And it is not via still a cell cycle function, but it regulates via a different pathway involving Wnt. So it may or may not be a proper cell cycle function of cyclin D, but it is for sure a CDK4 dependent that we have seen. But whether it's a cell cycle function or not, that we have not checked till now. But we are proposing that it could be due to the defect in the sheath cell development. And this is a indirect effect of sperm to oogenesis switch defect is due to the sheath cell defect.
Are there any other precedents for this cyclin D Wnt kind of interaction in other systems?
So sorry, I didn't get your question.
So are there any other instances where Wnt and cyclin D have this kind of regulatory role in other systems, in other developmental systems?
Okay, so I'm not sure about other organs like cyclin D and Wnt interaction. I have not checked. In other systems.
Yeah. I think in neural plate induction in the mouse, there's an interaction between Wnt and cyclin D. Okay.
Sounds good. You can look that up, Umanshi. You've got a lot of good feedback. Thank you all. Thank you very much. Well done, Umanshi. All the best.
Thank you.
And Umanshi, there are some questions for you in the chat window. You can address them after your talk.
Okay.
Okay, great. Okay, our next speaker is Akshaya. Akshaya, you want to start sharing your slides?
Yes, I will do.
Great. Excellent. Akshaya, we can see them. Their shape. We go to slideshow.
Is that fine now?
Yeah, great.
Okay.
Yes. Akshaya, we just don't have your video on. Do you not want to put your video on?
My laptop doesn't have that.
Okay, no
sorry,
problem.
camera is not working. Is that fine? Explaining my audio?
Yes, perfect. Absolutely. All the best.
Okay, thank you. Good afternoon, everyone. I am Akshaya Ashok, doing my PhD under Dr. Anjit Kumar at Manipal Institute of Regenerative Medicine. Coming to my study, which deals with the unconventional role of ubiquitin proteasome system in endodome cell fate specification. As all of us know that the protein homeostasis is majorly maintained by protein synthesis and protein degradation, but wherein protein degradation further accomplished by majorly two pathway lysosomal and ubiquitin proteasome system. But in eukaryotes, the majority of the intracellular protein degradation is taken care by ubiquitin proteasome system. Probably you might know what it is, but still I will brief you that ubiquitin proteasome system is a complex system where ubiquitin is a small molecule which initiates the process with the support of a cascade of enzyme activity, where these E3 ligase have the specificity towards the target protein to target them for the proteasome for the degradation. So that is why this traditional role of ubiquitin proteasome is well known as a degradation system from late 90s. But recently, studies have been shown that proteasome system having some untraditional roles like DNA repairment, immune response, etc. One among us, like a ubiquitin proteasome role, is extensively studied in pluripotent stem cells, differentiated cells. For example, you know that the reprogramming factors of SOS2, KLF4, CMYC, which are found that they are the ubiquitin proteasomal substrate by different E3 ligases. Not only the pluripotent stem cells, even the differentiated cells also found that key factors for the neurogenesis, hematopoiesis, so and so, they are going under the ubiquitin proteasomal system for its degradation. From this, it is evident that the ubiquitin proteasome system is playing major role in maintenance of pluripotent stem cells and differentiated cells. But one question remains unexplored is like, what is the role of ubiquitin proteasome system in cell fate? So this become fascinating us to look at further what is the role of ubiquitin proteasome system in cell fate. For that, we initially looked at the proteasomal activity during embryonic stem cells mediated cell fate specification. For that, we check the chymotryptic activity. The chymotryptic activity is very important for the protein degradation. To execute this, we use chymotryptic substrate, which is tagged with the AMC, which is a fluorophore, in presence of chymotryptic activity, which cleaves tyrosine and releases the AMC. And this fluorescence will be directly proportional to the change in proteasomal activity. With that, we performed a proteasomal activity as in targeted trilineage differentiated cells. And interestingly, we found that endodermal differentiated cells showed there is a significant decrease in proteasomal activity compared to other two lineages like mesoderm and ectoderm. Based on this, we had a question like, then what happens if you inhibit the proteasome with a proteasomal inhibitor, which is called MG132, during embryo body formation? Embryo bodies are nothing but they are the three-dimensional aggregates which compromises all the lineages. So based on that, we did a spontaneous embryo body differentiation in process of different concentration of the proteasomal inhibitor. And surprisingly, you can see here the endodome specific markers such as SOT17 and CHCR4 showed there is a dose-dependent enhancement in expression, whereas not mesoderm or ectoderm changed at all. And similar suppression we found with the Western analysis also. And this situation, we can have a question like, then what happens if you activate the proteasome? For that, we have done a embryo body differentiation in presence of proteasomal activator called IU1. And there we found that the endodome specific marker was not modulated much. If you see here in the Western blot analysis with the CSCR endodermal marker showed with the proteasomal activator, there is a diminished expression. Not only that, even we have done a trilineage differentiation in presence of proteasomal inhibitor. There also we found that the endodermal specific markers alone enhanced with the proteasomal inhibition, whereas the other lineages showed there is a significant decrease. So this observation is conclusively showing that proteasomal activity inhibition is specifically enhancing endodermal lineage. So this intrigued has to look at further. Then what happens if you inhibit the proteasome during mouse embryonic stem cells mediated pancreatic beta cell generation? For that, we have standardized their differentiation protocol from day 0 to 22. And then we checked we had induced the proteasome by day 3. And then we check the expression markers at a different stages of differentiation. And you can see here, compared to the differentiated control, there is a significant enhancement in expression of all the markers during differentiation. So this is informing that proteasomal inhibition is favoring beta cell generation. However, we want to validate this observation. For that, we used another model system called transgenic induced pluripotent stem cells. That reporter line we generated with the PDX1 GFP. So that is like whenever the PDX1 expresses, that will give a green fluorescence, and that will be indicating the PDX1 expression. With this, we also differentiated the PDX1 iPSC cells towards pancreatic progenitors, and we looked at the green fluorescence. With the differentiation, you can see here the enrichment of the green fluorescence. But if you inhibit the proteasome, that there you can see even more increasing green fluorescence. But we have not limited our study with a mouse model system. We explored in human iPSCs also. There also we differentiated human iPSCs towards the beta cell differentiation, and we looked at the expression of PDX1, which is a progenitor markers important for the beta cell differentiation. And you can see here with the proteasomal inhibitor, during differentiation, there is an enhancement in expression compared to the control. Not only PDX1, even SOX9, HNF6, these are the key markers required for the beta cell differentiation. Those also showed with the proteasomal inhibitor, there is a upregulation in expression. So all these informations are indicating that proteasomal inhibition is enhancing beta cell differentiation. But why? Why this enhanced beta cell differentiation happens due to proteasomal inhibition? That's become our next question. To address that, we have analyzed the temporal requirement of the proteasome during differentiation. For that, we induced the proteasomal inhibitor at the later stages of differentiation. And then we looked at the expression pattern of the key markers required for the beta cell differentiation. Surprisingly, we found that the enhanced expression was completely diminished with the later stages of inhibition. So this information is indicating that the early inhibition of the proteasome is only pushing the cells towards enhanced pancreatic beta cell differentiation, but not the later inhibition. So this gave us an idea that to look at the signaling pathway, which is highly involved in the early stages, not in the later stages. And that search, we ended up in identifying the Hippo-YAP pathway. So this Hippo-YAP pathway is recently reported in Nature paper, that which is essential in early endodermal differentiation, and this exit is also mandatory for the further beta cell differentiation. Keeping this in mind, we also checked the expression pattern of YAP during beta cell differentiation, and we found that YAP expression was enhanced at the early stages, and that was later on decreased, which was corroborating with the previous literature also. But we wanted to see then what happens if you inhibit the proteasome during beta cell differentiation, what happens to the YAP. And there also we found that with the proteasomal inhibition, there is an enhancement or upregulation of YAP expression at the early stages like day 6 and day 10. But if you see here day 16 at the mature stages of beta cell differentiation, there was no much change even after the proteasomal inhibition. And this data is indicating that probably the proteasomal inhibition during beta cell differentiation is stabilizing the YAP expression at the early stages. But we wanted to see what is the commitment of the YAP during beta cell differentiation. For that, we looked at the expression of multipotent progenitor markers. These are the key markers required for the beta cell differentiation. And there we used a YAP inhibitor, which is called vertiporfin. And then we looked at the SMAT6 SOX9 progenitor markers expression. You can see here with the YAP inhibition, there is a diminished expression. But when we simultaneously inhibit our YAP and proteasome, you can see here the restart of SMAT6 SOX9 multipotent progenitor markers. This is conclusively indicating that proteasomal inhibition is stabilizing the YAP pathway, and thereby which facilitating the enhanced multipotent progenitor markers. So the story so far we found that mouse embryonic stem cells mediated differentiation in presence of proteasomal inhibitor, specifically enhancing the endoderm lineages via regulating Hippo-YAP pathway, and that enhances the multipotent pancreatic progenitor, which in turn facilitates the better pancreatic beta cell differentiation. But one question remains elusive is like, but why this decreased proteasomal activity what we previously found in endodermal differentiation? Not only endodermal differentiation, if you see here, even the embryonic and adult pancreas showed there is a significant reduction in chymotryptic activity. So this is indicating that inherently the endodermal differentiation having lesser proteasomal activity. But what could be the reason? So to evaluate that, we looked at the contribution of the ubiquitin proteasome. So basically, this ubiquitin proteasome is composed of two particles. One is 20S core and another one is 19S regulatory particle. Where this 20S core particle does the proteolysis process and the 19S regulatory particle helps this process via opening this gate entry. So we wanted to look at the contribution of 20S core and 19S regulatory particle activity separately. For that, we used a different approach by using SDS, which is a detergent. And in presence of SDS, if you are looking at the activity, that will helps to open up this gate for the protein degradation without the support of 19S regulatory particle. So whatever the changes happens in presence of SDS, that is indicating 2DS core activity. If there is no change at all, then that implies 19S regulatory particle activity. With this, we decided to differentiate the cells towards pancreatic beta cells. And during the differentiation, you can see here, there is a significant reduction in proteasomal activity. And when we have done the proteasomal activity in presence of SDS to see the 20S core activity, there was no change at all. So that is indicating whatever the decrease we found is here, it is probably due to the 19S regulatory particle. Thus, we decided to look at it. Then what is the contribution of 19S regulatory particle during endodermal differentiation? Surprisingly, there we found a key candidate PSMD2 was significantly down-modulated during endodermal differentiation. But we wanted to see whether this down modulation is uniform in all the trilineages. To see that, we looked at the trilineage differentiated cells, the PSMD2 expression. There also we found that yes, PSMD2 down modulation is particularly in endodermal differentiation at a transcript and protein level. Not only this, we also checked the PSMD2 expression tissues. That also showed pancreas is having the most down-modulated PSMD2 expression compared to other tissues, which is representative of the other lineages. So this data collectively indicating that PSMD2 is the key candidate which is drastically down-modulated in endodermal differentiated cells, and that would be one of the possibility in what we found that endodermal differentiation, there is a decreased proteasomal activity. But we wanted to see how this PSMD2 regulates during endodermal differentiation. For that, we looked at the key mechanism that is regulating PSMD2 in endodermal differentiation. For that, we used a small molecule mediated inhibition of the key signaling pathway required for the endodermal differentiation. And there we found that cyclosporin A, which is the calcineurin inhibitor, is rescued the PSMD2 expression than any other pathway. This is indicating that cyclosporin A mediated abrogation of calcineurin NFATC pathways rescuing PSMD2 expression in endodermal differentiation. What is the contribution of calcineurin NFATC pathway in endodermal differentiation? Yes, that would be a possible question. So we also asked the same question to see the importance of the calcineurin NFATC pathway. We did a differentiation in presence and absence of cyclosporin, which is a calcineurin inhibitor, and then looked at the HNF6 sortine, which are the key markers required for the endodermal differentiation. And there we found that HNF6 sortine, where the key markers showed with the cyclosporin inhibition, there is a hindrance of HNF6 sortine markers. Whereas if you see here PSMD2, which showed with the cyclosporin A, there is an enhancement in expression. Even transcript analysis showed the similar effect. So that is indicating that calcineurin NFATC pathway is crucial for endodermal differentiation, and whenever the cyclosporin activates, that means the calcineurin NFATC pathway inhibits, that is simultaneously enhancing PSMD2 expression on contrast, which is inhibiting the endodermal differentiation. So we wanted to see this calcineurin NFATC families actually consist of different classic members, but among that, which classic member is highly regulating the PSMD2 expression. To see that, we had done a nuclear extraction of mouse embryonic stem cells and endodermal differentiated cells, where we found that NFATC1 is the key candidate was enhanced during endodermal differentiation. So this is indicating that NFATC1 is the key candidate is present in beta cell development, highly modulated. So then we wanted to see then what happens if you activate the NFATC1 with respect to PSMD2 expression. For that, we generate a NFATC1 overexpression construct and then looked at the PSMD2 expression. As expected, we found that with the NFATC1 overexpression, PSMD2 expression was drastically down modulated. Even in the protein analysis also showed the same. However, we wanted to confirm this observation very keen. For that, we have analyzed the PSMD2 luciferase activity at the promoter level. Then we were in the presence of different concentration of NFATC1 overexpression. And you can see here with the increasing concentration of NFATC1, the PSMD2 promoter activity was significantly down modulated. And this observation collectively indicating that NFATC1 is the repressor of PSMD2 expression at endodermal differentiation. So, however, this information is indicating a key point that calcineurin NFATC pathway and ubiquitin proteasomal system having a cross-link in endodermal differentiation via linking a PSMD2 candidate. The story so far, we found that calcineurin NFATC pathway is important for beta cell differentiation, and the NFATC1, which is the downstream target, is suppressing the PSMD2 expression, and that is the reason we found that decreased expression of PSMD during endodermal differentiation, and that promotes the further endodermal differentiation. However, we found that the loss of activity of PSMD2 in endodermal differentiation. So we were so curious to see then what happens, the PSMD2 role in endodermal differentiation. To see that, we performed a loss and gain of function by generating two different constructs. One is PSMD2 knockdown, the another one is PSMD2 overexpression construct. And first we validate the efficiency of the construct. Then we had done a spontaneous embryo body differentiation in presence of PSMD2 knockdown cells. And there also, surprisingly, we found that endoderm differentiated cells, that HCHCR sortsafindine are the key markers for the endoderm, showed there is an enhancement in expression than any other lineages. So this is indicating that PSMD2 shRNA is giving the favor towards endodermal differentiation. However, to confirm that observation, we have done an endodermal differentiation towards pancreatic beta cells. Then we looked at the SOX9 and HNF6 are the key markers during beta cell differentiation in presence of differentiated and PSMD2 shRNA mediated differentiated cells. You can see here upon PSMD2 knockdown, there is an enhancement in expression of HNF6 and SOX9. Totally that even the PDX1, which is the key marker, showed there is an upregulation upon knockdown condition. But when you overexpress on the background of shRNA, which showed a diminished effect. So that is showing that PSMD2 shRNA is specifically enhancing the endodermal differentiation. So this became very curious to us, like rather than a ubiquitin proteasomal subunit playing its traditional role, the PSMD2, which is a UPS candidate, is having an unconventional role in endoderm fate specification. So, however, we wanted to further see whether if the calcineurin pathway is upstreamly regulated the endodermal differentiation, then what would be the downstream mechanism of PSMD2 in endoderm specification. For that, we had done an in silico analysis, and for the protein-protein interaction, there we found a unique combination of PSMD2 nanog interaction with respect to GATA4. And further, we wanted to check, it was previously studied that nanog is the repressor of GATA4 and which terminates the endodermal differentiation. So further, we wanted to see anyhow whether this PSMD2 and nanog interacts each other or not. For that, we have done a immunoprecipitation. For that, we used nanog and PSMD2, which is tagged with the flag and HA respectively. Further, we pulled down the flag and then looked at the nanog expression, and you can see here, as good as the input, there is a strong interaction of PSMD2 and nanog at the downstream level. So this was a very interesting observation. So then we decided to look at, then if this is the criteria, then what happens with the nanog and GATA4 interacting. For that, we performed a nanochip assay in presence of PSMD2 shRNA and overexpression. Then we looked at the GATA6, GATA4 expression, which are the key markers required for the in early endodermal differentiation. If you see here in the shRNA condition, the nanog lost its binding affinity towards GATA6
Akshaya,
and the
sorry
hello.
to interrupt, but 20 minutes are up. Could you
Yeah,
stop?
I am about to finish, ma'am.
Sure, sure.
Okay, so we here it is lost PSMD2 shRNA. Sorry, nanog is lost its binding affinity towards GATA6 and it lost its interaction. Whereas the PSMD2 overexpression enhances nano GATA6 interaction. This is showing that GATA6 and GATA4 are the intermediate candidate of PSMD2 nano GATA6. To have a holistic view of my study, here we are addressing a calcineurin NF-ATC pathway is the key pathway in endodermal differentiation, where its downstream target NF-ATC1 is terminating PSMD2 expression and thereby which enhances the endoderm differentiation. But in case of PSMD2 and nano interacts each other and which act as a gatekeeper for endoderm genes and thereby which is terminating the endodermal differentiation. So this is a novel information where the calcineurin, NF-ATC and UPS work each other for the endoderm fate specification via a ubiquitin proteasomal subunit PSMD2. So if you are considering this as an application level, you know that the cyclosporin is an immunosuppressant that usually used for the organ transplantation. But recently people showed that 70 to 80% of the people are going under the post-transplantation diabetes due to the organ transplantation. So we hypothesize that probably the cyclosporin A usage upregulating the PSMD2 expression, thereby which is hindering the pancreatic beta cell development. Probably that would be the reason behind the enhanced diabetes condition. If this is the criteria, targeting a PSMD2 via small molecule mediated approach will be a potential approach to rescue the post-transplanted diabetic condition. So with this, I would like to show my great gratitude towards India Bioscience and the company of biologists for providing a great platform to great platform, and the Manipal Institute of Higher Education and funding source of SERB DST, and of course to my lab and my great mentor Professor Anujit Kumar and my cheerful lab mates. They were always with me for everything, and thank you. With this, I would like to have a query. I would like to take a query.
Sounds good, Akshaya. Thank you so much. All right, let's take question and answers. Surendra, Jyotsna, Prasad, Raj, please go ahead.
Maybe Raj can start now.
Sure, Raj, go ahead.
Okay. Akshaya, that was lovely. Did you look at YAP localization as well?
Oh, sorry, I couldn't hear you. 1 second, could you repeat it?
Did you look at YAP localization?
We haven't looked at the YAP localization, so unfortunately, we would like to look at whether it is nuclear localized or not.
Okay. Okay. Okay, I'll pass on to.
Yes, sure.
Sure, great.
Yeah.
Anyone else?
I can ask a question here. So Akshaya,
ma'am.
very nice, very comprehensive study.
Oh, thanks,
Yeah,
ma'am.
I just to make sure that I understood your complex schematics and pathway. What you're showing is that basically the suppression of endodermal differentiation, early endoderm differentiation by this particular proteasomal subunit, the regulatory subunit PSMD2, is what is kind of keeping a lid on a cap on endodermal differentiation. And that when you release that, you actually have a more enhanced endodermal differentiation, right?
Yes.
And what you're showing is that you think that PSMD2 is doing this through its function of keeping nanog activity low.
Yes.
Right?
Yes.
So now what confused me was the picture that you had in the nucleus, where you had PSMD2 and nanog interacting.
Yes, ma'am.
And you seemed to indicate that this was a regulatory interaction that somehow was affecting the gene expression. Whereas what I understood from your talk is that actually PSMD2 is increasing the turnover of nanog protein, and that when you abrogate PSMD2 expression, you actually have a higher level of nanog. Is that the correct interpretation, or am I wrong?
Yes, ma'am, partially, but I would like to tell that one thing is like whatever the change we found with the PSMD2, we are not claiming it is doing as a part of ubiquitin proteasome. That's why I mentioned a word which is showing an unconventional role than its role.
But what evidence do you have that it's an unconventional role? Have you actually localized it with respect to the proteasome itself? I mean, how do you know it's not functioning through the proteasome?
Yeah, actually, this is a part of study we wanted to explore further. So that's why I didn't show any much result over here.
Okay.
Yes, we are suspecting that the PSMD2 is not much doing its the specific role as a component of ubiquitin proteasome. However, we are actually doing a process to screen whether the PSMD2 is nuclear localized or cytoplasmic. What is the localization of PSMD2 in fact?
I mean, wherever it is, because even there are proteasomal subunits, and proteasome may be active even in the nucleus, right? So
yes,
the
of course,
question
yes.
is, you have to be able to distinguish. I think this
Yes.
is going to be key for you. You have to
Yes.
distinguish whether it is acting as part of its proteasomal functions
Or
if
alone.
it is something else. Because
Yes,
in
yes.
any case, you're still seeing a reduction in the protein amount, right? So it could
Exactly,
still be
ma'am.
because of degradation.
Yes.
So
yeah, we have to study further for that, whether it is a part of UPS or it is doing alone. That have to be further, we have to do pursue.
Yeah, can I go ahead? Yeah. So along the same line, Akshaya, I just
Hi,
wanted
sir.
to know the PSDM2 level is going down, right? What you said.
Yes,
So,
yes, PSMD2.
So, but you think it still has some role to play in this differentiation process, like endodermal lineage differentiation?
Yes.
Is it possible that if I have a hypothesis, the subunits of your proteasome itself is getting modulated, changed? There are some other subunits coming up, and that's how they are driving the endodermal differentiation. Is it possible?
Actually, in fact, sir, your good question. But we have screened all the ubiquitin proteasomal subunit with respect to the endodermal differentiation, and we found that PSMD2 is the key candidate, is highly modulated in particular aspect of the endodermal differentiation. So I can't claim that is there any other component is doing something perfectly, and that is the consequence where PSMD2 suppresses endodermal differentiation.
Okay, so in that case, what do you think? How does the PSMD2 is directly targeted?
That's what it is, sir. We have to further look at a in-depth study whether the PSMD2 is doing this specific role and along with the factor of UPS component, or it is doing specifically alone, as which is freely available in cells, so that is doing alone this function. So that we have to cross-check via localization.
Okay, and one generic question: you
Yes,
could
sir.
see only difference in the differentiation. There is a commitment towards the endodermal lineage in the when you perturb proteasomal, like when you do a proteasomal inhibition in the early stages of
Yes.
differentiation, right? But you do not see any changes in the proliferation, survival, anything else.
Yes, actually, we had the proliferation per se with respect to proteasomal inhibition, whether it is modulated or not. And we found there was no much modulation with respect to proliferation. So I can tell something is doing with the proliferation. That's why the change we found in endodermal differentiation upon proteasomal inhibition. So there was no much modulation.
Yeah. So when you say lineage is particularly skewed to the endodermal side, does it mean the percentage is increased, or is like 100% skewed to that lineage?
I can say probably which is favoring endodermal differentiation than any other lineages. So probably those markers are not getting enhanced, and whereas the endodermal differentiated markers are highly gets enhanced, probably those markers will helps towards the endodermal lineage only. Probably that would be the fact happening inside.
Okay, good. Okay, I'm done.
Thank you, sir.
May I ask one question at the end?
Sure, sir.
Yeah, you mentioned that your work may have some application in people who are prescribed with cyclosporine in organ transplant.
Yes, sir.
What do you mean, application?
Yes, actually, sir, recently report has come like a cyclosporine A, which is a immunosuppressant drug that actually inhibits the calcineurin NFATC pathway. So the people who are went for the transplantation, nearly 80% of the patients reached in post-transplantation diabetes. So we are suspecting that probably it will be due to the cyclosporine A usage. That means the calcineurin NFATC inhibition. If that is the case, we are hypothesizing that PSMD2 expression probably gets enhanced in that criteria with the calcineurin NFATC inhibition. If
That
is
I
the
understand. That I understand. But how do you foresee application?
Yes, sir. If that is the criteria in post-transplantation diabetes case, we can use as a PSMD2 small molecules target. So if we are developing a small molecules to target PSMD2 in specific condition, that would be a great approach to reduce the diabetic condition.
Okay. By the way, there are many excellent, substitutes for cyclosporine now, like tacrolimus and many others. But all of them tend to induce hyperglycemia in post-transplant cases. So maybe you can look at their mechanisms also. There are quite a few of them.
Actually,
Yeah.
sir, what you mentioned is also correct, but the FK506, that is the tacrolimus, that is also inhibiting calcineurin NFATC pathway only. If we are looking at that mechanism also similar way, it is inhibiting calcineurin NFATC pathway. So it is both in hand same only.
So that makes it more important to see whether such treatment will be safe at all.
Yes, sir. Actually, small molecules are in practice in animals models. So we wanted to further see how efficient in clinical practice. Actually, in pancreatic cancer also small molecules are available. If this is the criteria, we can look a chance that these small molecules can be used for diabetes.
Because I might add that post-transplant patients are managed by clinicians very, very carefully. And as it is, there are a lot of problems there. So you might want to look at that aspect also. Okay.
Oh, sure,
Okay.
sir. Yeah,
Thank
further
you.
investigation is needed. Yes. Thank you, sir.
Thank you, Akshaya. Thank you, all the expert mentors. We can move to our next speaker. Akshaya, you can stop sharing. So Vishwadeep is our next speaker. Vishwadeep, could you please confirm with your audio and video?
Yeah, am I audible?
Yeah, you are audible and visible. Please start sharing your slides.
Yeah, just a minute. Is my screen visible?
Yes.
And in the PowerPoint mode, I suppose.
Okay, go ahead. 20 minutes.
Yes. Hi, everyone. This is Vishwadeep. So I'll be talking about how plants make the robust organ shape and the regular flat structure. So this is one of my PhD thesis, one of the chapter of my PhD thesis. So when we talk about plant organs, you have a remarkable diversity of shapes and sizes. Now, the interesting question is, what is the biological basis of this diversity? To biologists, many of them argue that it comes from the genetics, which is very true. But then is it the only cue that makes this kind of diversity is the question that we are trying to address. Now, when we are trying to address this question, we have to go a little bit deeper into what exactly is happening over here. So when we take an organ over here, for example, this is a plant organ. And if you take an individual cell of this plant organ, it has got genetics and it has got a lot of biochemical networks inside a particular cell. And this is your input, which is happening at the cellular level. But then your output is a multicellular organism or an organ here in case this is a cotyledon or a plant embryonic leaf. And irrespective of what is happening at the cellular level, you always get this reproducible shape and size. So this is your input, which is at the cellular level and a single cell level, and the output is this. How can biology or the genetics actually make a geometrical output such as this? So it's not enough to explain that. That's where the power of mechanics comes into the picture. So what do I mean by mechanics? Mechanics is basically the study of the geometry. Here in this case, we are talking about the geometry of the cells as well as the organ. And this geometry is also coming into the picture because of the mechanical forces which are acting upon the organism, as well as it comes from within the cell as well. And somehow at a bigger level, all these feedbacks merge together in order to make a robust organ shape. Now, when we talk about mechanics in plants, so we need to understand some few terminologies, and we should ask ourselves first question. There is some commonality between plants, bubbles, and the balloons. So taking a balloon analogy, so if you draw a balloon, if you draw a square on the balloon and inflate it with air, with the pressure, it inflates and you see that the square increases in size as the balloon is increasing in size. And the pressure is uniform over here. That is why the square is increasing uniformly. Now, if you take a balloon, which is of not a uniform shape, and you draw the same thing, and then you increase it over the course of time, you see that because of the distribution of the pressure and the geometry of that object, the pressure is distributed unevenly, and the square that we drew is expanded in a different fashion. So what is happening over here? Due to the air pressure, the pressure exerts force onto the area. That is what we call a stress. And then this stress eventually leads to growth. And that's what we are seeing, that the balloon is increasing in size. Now here in the case, it's a isotropic stress and uniform growth. And here you see it's an anisotropic stress and a directional growth. Now, how to maintain this structure? You need to have a balance between the inner forces and the outer forces. And that's how the structure is maintained. If you have it in a larger quantity, it cannot maintain itself and it bursts off. Similar thing exists in plants. Each and every cell is having a turgor pressure, which is maintained by the water. Now, that's what exactly happens in plants. Plants behave like balloons. So the cell wall of the plants is under the turgor pressure, and there is a constant influx of water inside and outside of the cell. And when the hydraulic pressure and the turgor pressure exceeds a certain value, which is we denote it by the threshold, that's when the cell starts to expand or grow. And then obviously the question is, why don't the plant cells burst and how do they mitigate the stress? Because the pressure is actually in the range of 10 megapascal, which is enough to crush your bones. So plants actually have a remarkable ability of understanding this mechanical stresses. So for example, if you take here a cell which is cylindrical in shape, by virtue of its shape itself, it has got its maximal stress in the transverse direction and minimal stress in the axial direction. Somehow plants understand this, and the microtubules, the cortical microtubules, align to the maximal stress. And what they do is actually bring the cellulose in that direction to avoid the bursting. And that is why the growth usually happens in the direction of the minimal stress. A lot of people argue the fact that plants grow only because of the sunlight in the upper direction, but there is a mechanical basis to it as well. It's very easier to grow in the upper direction because you have a lot of forces, mechanical stresses occurring onto the transverse direction. So this brings me to the mechanical feedback loop, which occurs in plants. So if you have a shape of a cell, which is growing now, it's having its direction, and that shape is prescribing some direction of the mechanical stress. This mechanical stress is understood by the microtubules, and then they will curb the growth of that cell. Now, plant cells are linked by cell wall with each other. So you need to understand the growth of each and every cell in order to grow into a proper and robust organ. Now, the question is, how does this mechanical feedback helps in making robust organ shapes and sizes? So a lot of experiments have been done to understand what exactly is happening over here. So mechanical feedback, like I said, is necessary for maintaining the robustness. What do I mean by that? So suppose you have an organ over here, and because of the cell wall, you're linked to each and every cell, and you need to understand the growth of each and every cell. Now, if I say that the cell here in the center is growing more faster than the other cells need to understand that it is growing faster, and they need to actually understand the mechanical stress over there. So here what is happening is the cells adjoining this red cell are aligning the microtubules in this direction because the stress is more in this direction. This is how they will reinforce with cellulose, and so that this cell will not crush them. And that is where this mechanical feedback buffers this growth heterogeneity what occurs in the cellular level, and it makes the robust shape. Unlike animal cells, the plant cells cannot move. So you need to have this buffering of the growth heterogeneity. Now, what if you don't understand this? So what if you don't understand the growth of one or the other cell? So you do not have this proper mechanical feedback, you cannot understand this, and you'll have variable shape and size of the organ. And that brings me to the question that how can we incorporate the mechanics into the developmental biology or understanding how robust shapes are formed? Apart from genetics, we are incorporating the mechanics. And how do we understand what mechanics is actually doing over here? So like I said in the beginning, you have the input, which is the biochemical patterning at the cellular level, and your output is a reproducible shape and size, which is a multicellular organ. So what we essentially do is to understand how are the growth patterns occurring over there, and with the help of live imaging, quantitative live imaging and modeling, we try to understand that how genes or the genes occurring in a particular wild type or a mutant affect these growth patterns. And then we model, and then we can say that these are the genes which are necessary for redistribution of the growth patterns. And inherently, some or the other way, we can understand the mechanical forces which are related to this. And then we can give a bit of an holistic idea about how a reproducible shape and size of the organ is formed. So that brings me to my case study, which is cotyledon. So why cotyledon? So in plants, you have multiple organs. So leaf and sepal are similar kind of organs. They are leaf-like organs. And of course, cotyledon is also a leaf-like organ. But these are very distinct classes of organs because leaf and sepal are a mixture of proliferation and expansion. Now, interestingly, in plants, you have division of the cells, and then they go into expansion, which is nothing but differentiation. The ploidy increases and the size of the cell increases. There are only very specific cells which are differentiated, like stomata or trichome, which is like a hair-like structure, but majority of the cells just go into expansion, that is increase their ploidy and increase their size or volume. Now, interestingly, cotyledon is also a leaf-like organ, like I said, but then the thought of school in botany says that cotyledon has a distinct phase of growth. One, which is pre-germination, when the seed is being formed, it is proliferating, and post-germination, it just expands. There is no division at all. So that is what the school of thought in botany goes around. So we wanted to understand, is it really so? And we wanted to ask two main objectives. One, how is the wild type growing? And does it have any proliferatory mechanisms post-germination? And does it follow a typical gradient of growth? So in case of plants, what happens is they follow a gradient of growth. So it can be proximodistal, it can be mediolateral. So what I'm showing over here is, if you see over the course of time as the organ, this is the leaf, it grows over the course of time. In the earlier stages, the red color indicates it grows faster. But as the plant is aging, you see that the region at the tip becomes blue in color. That means the tip is starting to stop its growth, and slowly this growth goes down. And that is what I mean by basipetal gradient of growth. Now, this typical basipetal gradient of growth is observed in case of leaf and leaf-like organs, which is leaf and a true leaf and a sepal, because they are proliferating and going into expansion. But whereas the thought of school in botany says that this is separate, so we wanted to test that whether it is really true or not. So how do we do that? We test exactly what is happening in the wild type, because there is no whatsoever report on how does a wild type cotyledon in Arabidopsis actually is formed through quantitative live imaging. And we want to test the robustness of the growth patterns by working with genes, genetic models which perturb this, and by perturbing the environmental modules like light. How do they affect the shape and size of the organ along with the surface curvature? So to give you an idea about, this is a wild type cotyledon imaged over the course of 10 days. So you'll see that it grows over the course of time. It's not a big organ as such. It just helps in the kickstart of the life of the plant. So when we see as it grows over the course of time, the area reaches a plateau of around 3.3 millimeters square, which is fair enough for a wild type. And if you see, it's mostly a circular organ. To quantify, we actually calculate the isotropy of this, which is nothing but the length by width ratio. You see that as in the beginning, it's more anisotropic, and as it progresses its growth, it becomes more and more isotropic or circular. Interestingly, if you'll see, these plants are also having an inherent curvature, as I'm pointing out my laser over here. So this curvature we essentially see only in the plate, which is grown in an artificial, chamber. We do not see this kind of curvature actually occurring in plants which are growing right on the soil. So, which is interesting for us to understand why is it happening. And like I said, we tweak the genetics. So we take use of one important gene which maintains the transition of the cells from proliferation to expansion. It's called as TCP. So this is a mutant wherein the TCPs are absent, and you see that this mutant has the cotyledon which grows only in the proximodistal axis. So if you see, the area is actually 1.5 times bigger than that of the wild type, and the length by width ratio is actually increasing. That basically tells that it is growing in an anisotropic manner instead of an isotropic manner. So why is it exactly happening? So like I said, we actually do the live imaging. We take the same cotyledon and then image it over the course of time. So these lines are the plants that we are looking at. These are labeled with the plasma membrane tag. So this plasma membrane is tagged with the GFP. And since the turgor pressure is extended onto the plasma membrane, it's almost near to the cell walls, and that's how you can quantify the geometry of the each and every cell and how is the organ growing over the course of time. So this is how a wild type looks like. This is 48 hours post keeping it in the light. And this is the mutant that I'm talking about. So this is how pretty much it does not look so much different in the earlier stages. I'll come to that, what changes are occurring over here. So, but when we take a side view of this cotyledon, as it opens up over the course of time, you see that it is a relatively flat organ in case of wild type. But that does not occur really in the case of the mutant. In mutant, it starts to curl up. It has an inherent curvature. So what we essentially do is we try to take the image of each and every cell of that cotyledon at the particular day and then map it out. How is it growing over the course of time? So here on the slide, you're showing a map, a growth map. So this is the color codes indicate absolute area increase of each and every cell. It can be through division or also it can be through expansion. So if you see that in the first 12 hours, 48 to 60 hours, it does not really grow more. However, the edges are growing a bit. So you see that the increase is not much in the 48 to 60 hours, but suddenly at 60 to 72 hours, it starts growing a lot. And the growth is more or less limited to the lamina of the organ. And later on, you see in the next time point, you see that the growth is more or less towards the center part of the lamina. So it wants to make the organ circular. So it is trying to shift its growth onto the mediolateral axis rather than the proximodistal axis. And we start the imaging at like 3 or 4 in the morning. And so every 12 hours, we image the cotyledon. And you see that it grows mostly during the night time. It does not really grow much during the daytime. So this is what a wild type looks like. So now the question was, what exactly is happening with the cell division? If you remember, I told you that there is no division in the cotyledon that was reported, and the last report is 30 years back. So when we did the live imaging and we actually quantified that indeed, actually there is a division of cells. Most of these cells, which you're seeing over here at the 60 hours, they're actually dividing. So the scale, the heat map basically tells you what is the number of divisions over the course of time. So you see that 40% of the cells are actually going under division, single division from 48 to 60 hours. And then later on, this division continues. And there are certain cells which go into 2 or 3 divisions. And these 2 or 3 divisions basically contribute to number of stomata. So for the first time, we are seeing that the cells actually divide post-germination. Now, interestingly, this is what happens in the wild type. So we wanted to understand what exactly is happening in the mutant. Does it behave similarly, and why is it growing only in one axis, and how come the curvature comes up? So when we see the mutant, you see that in the first 12 hours, you see that the growth is actually more in the margin. This is the wild type, and this is the mutant. In the mutant, you see that the growth is more in the edges of the mutant. And then just like the wild type, it grows more in the 72 hours. But here the growth is more redistributed, unlike that of the wild type. In the wild type, it's more centered towards the lamina. Here, everything is growing. And then you see in the next time point, you see that the growth is shifting towards the base. The tip is already going into expansion, that it is not growing. And that's how we think that the curvature comes up. But then when you look at from an organ perspective, you see that there is not much difference into the growth patterns when you see the organ per se. So we wanted to understand how and why, what exactly is happening over there. Now coming to the cell division pattern as well. So this is what is in the wild type. So in the mutant, you see that it has actually more number of division. If you remember the mutant that I was talking about, these genes are responsible for making the cells proliferate more instead of going into expansion. So here we see the same result. So in the mutant, the division is more, and the division is almost, there are more cells going into 2 and 3 divisions in the first 12 hours, and then later on it subsides just like the wild type. And so then we ask, so more or less the growth pattern is similar in the wild type and the mutant, then how come we get the curvature and a different organ shape altogether? So for that, what we essentially do is, so you have each and every cell. So we want to see how each and every cell in the next time point is growing across the proximodistal axis and the mediolateral axis. So this is first the map of the proximodistal axis. So what we essentially do is we take the cotyledon, we divide it into 5 regions, and then at a particular time point, we see which region is growing at what rate. So here you see on the left, the wild type. So, and this is the growth across the proximodistal axis in terms of percentage. So this is the distance from the tip, and this is the base. So you see that it's growing uniformly in the first 12 hours. And then all of a sudden, the center part of the cotyledon starts to grow more. That's what you see over here. And then the base is not growing more. And then in the next time point, you see that the tip is not growing more, whereas the center part is growing more because it wants to create a circular lamina. So it is shifting its growth more and more towards the mediolateral axis. And then you see that as it tries to unfold and open itself, so there is a region where it does not grow more, and that's where it unfolds. And that's what is seen in the graph as well. So unlike in the mutant, the growth patterns are redistributed actually. So if you see in the mutant, the similar kind of graph, you see that the tip is growing more and the center is not growing more. And again, the base is growing more. Then over the course of time, what happens is the growth patterns are redistributed over here, wherein the tip is growing more compared to that of the wild type. And then you see that it tries to grow more in the base because it wants to establish that curvature already. And then in the next time point, you see that the upper part is growing more because it wants to extend that upper part in order to make that curvature. So a fine distribution across the proximodistal axis is enough to explain the curvature, what is happening. Overall, the growth does not look different, but when we dissect the growth into such patterns, we actually see that what exactly is happening. We do the similar thing from the mediolateral axis. So what we essentially do is over here, so in the proximodistal axis, the tip and the base, here we take the center, the midrib as the center, and from that towards the edge, and we divide it into 5 regions, and then we see how is the growth being redistributed over the course of time. So in the mediolateral growth, you see that the center part of the cotyledon is not growing more, but the edges grow, which is very evident in case of the heat map as well. Over the course of time, the center part grows more, and the edges are also growing more because it wants to make a circular lamina. But then at some point, the edges stop growing in the next time point. Why? Because you have already made that framework, which is now enough. And then you can just start to grow in the center part in order to make the lamina circular. Now,
sorry to interrupt, Vishwadeep. Your 20-minute time is up. You can wrap.
Yeah, sure. So in the mutant, this growth pattern is more or less similar in the mediolateral axis, a little bit more redistributed. So we wanted to also understand how, apart from genetics, how does light modulates the surface curvature. So I'm just giving you a glimpse of what exactly is happening. So we grow these cotyledons in a setup wherein the light is only coming from a particular direction, and you see that the wild type becomes more and more flat compared to what you have seen in the earlier slides. Whereas the mutant, which we were to our surprise, is actually also losing its curvature. So genetics being a strong hand at maintaining the growth patterns and the curvature is also losing. So it looks like it's an interaction between the environment as well as the genetics in order to maintain the robust organ shape and implementing the curvature over here. So summary of my talk, more or less the overall growth patterns are conserved in the wild type and the mutant, but the wild type mutant shows proliferation in the earlier stages of growth, which is for the first time we are reporting. And then the curvature can be explained by the differential redistribution of the growth patterns across the proximodistal and mediolateral axis. So with this, I conclude my talk, and thanks to my lab. And this is a collaborative project between two teams, which I'm a part of in Canada and in France. Thank you very much.
Thank you, Vishwadeep. It was an interesting talk. We can move to our expert mentors. It's quite over the time, so we can have Q&A till 3:46, 47.
Okay.
Okay, Vishwadeep, you mentioned some genes that control this expansion,
Yes.
proliferation versus expansion. So, are these genes identified, and whether they are how they are regulated? Whether there are gradients of signals there?
So the thing is, these genes have been identified since the past 20 years, and a group in John Innes Center and Caltech, they identified these genes. These genes are actually belonging to a class of a basic helix loop helix transcription factor. What they basically do is they interact with a lot of cyclines and kinases, and to do their job, they stand at the checkpoint of proliferation. Wherever this gene is being expressed, it stops proliferation, and the cells immediately goes on into differentiation. So that's what we know of. With respect to the gradient, of course, we know that these genes are expressed exactly at the boundary of the proliferation and differentiation. So like I said, in leaf and leaf-like organs, where you have a lot of cells which are dividing, so where you express this gene, they go straight away into differentiation. And we see that this gradient is perturbed. But we do not know whatsoever what is exactly happening in cotyledon, because no study has been done on cotyledons per se. So we do not know whether the gradient was there to begin with.
So you don't know the signals that control their expression, or whether light has any role to play there?
So we know for sure that at least with respect to these genes, they are expressed everywhere in the cotyledon. Now the question is, how do they do it? We do not know. We stumbled upon this serendipity wherein we just grew the plates we grew the seeds into the soil, and then we see that the curvature is almost lost. So we think that it is a light-based phenomena because the seed is deep inside the soil, whereas in the plate, it gets light from all the angles, which might be controlling the curvature.
Okay. Interesting work. Thank you.
Thank you.
So very interesting topic, Vishwadeep, and nice presentation. I was wondering, here you're looking at the implications of altered proliferation and the switch from proliferation to expansion in impacting the mechanical properties of the. What about if you were to actually directly impact the mechanical properties of the cells by using mutants where you would expect to see changes in the stiffness of the cells or in the cellulose framework or in any of those? How does that impact the growth?
Yeah.
Does it actually impact proliferation itself?
Yeah. I'll come to that. So what we have seen, at least the genes that we work on in our lab, the genes that I have mentioned right now and the mutant that we are working on. So we have seen that actually in the leaf, it has got a very beautiful phenotype. The leaf is usually a flat and a circular structure in case of Arabidopsis, but when these genes are absent, it becomes frilled. It's like a kale. So we know what is the base mechanism going on, excess proliferation, which is causing this. But then essentially the frilling occurs because there is a mechanical conflict between the cells which are going into differentiation and proliferation. And that's where it buckles, and then in order to accommodate that stress. So we know that for sure that our genes are somewhere down the line impacting the mechanical stress patterns, and how they are understanding that we do not know. Now coming to the second part, we know of some mutants. So if I said you that the cell walls are linked together, so we have some mutants wherein the adhesive forces or the cell walls which are for example, like I said, the microtubules which are responding to this stress. So we have mutants wherein the response to the stress is hyper as well as hypo, and there we actually see that it actually does an effect on to the patterning of the growth as well as the overall organ morphology as well. So we know that what exactly is happening down the line.
Thank you.
Can I follow up
Sure.
the microtubule stuff? These hypo and hyper mutants. How do they affect kind of the stiffness of the microtubules and the integration with the cell? And you'd expect kind of supracellular organization as well of microtubules.
Yes, yes.
To pattern the forces across multiple cells.
Yeah.
How is that integrated?
Yeah, so if you remember, I was talking about the mechanical feedback wherein the shape is prescribing the mechanical forces over here. So now what happens in the mutants wherein we are having an hyper sensitive response to the stress patterns which are occurring. So these cells will not actually grow because the cellulose is being reinforced in that particular direction. And that is where the integration at the cellular level is translated to the organ level as well. So the final organ shape over there is actually small because the cells have not got any capacity to expand as their typical framework suggests. So that is what happens in these mutants. And we have seen that when the cells are unable to identify or understand the microtube, the stress patterning, you see that the organ just basically expands. And in some cases, when you depolymerize the microtubules, the cells basically explode like soap bubbles.
Very nice.
Okay, so which was just one general question. You said mechanics matters, genetics matters, right?
Yeah.
All of these things will matter for the patterning and orientation and everything. So is there any hierarchy in this? Do you think something?
So I think the most important part over here is unlike animal cells, plant cells cannot move. So actually they have involved a lot of abilities to understand the mechanical patterns. For example, if you see, why do you have coconut trees only onto the the shores of the sea? Because those cells, I mean, the leaves are blade-like structure which can actually withstand the winds which are coming through the sea. So I think there is no hierarchy as such because the plants have evolved a structure wherein to understand the mechanical patterning because you cannot move. In fact, if you take a very basic example, if you don't water your plant, you see that it wilts. They grow, in fact, under the pressure. So there won't be any hierarchy over here because all of these patterns are collectively responsible for understanding stress externally and internally. And I don't think so there will be any hierarchy over here.
Okay, just one quick question. In the mutants, you could see a lot of cell division, right?
Yes.
So this cell division, were the cells were same size as they were in a control, or was there any difference in the size?
So with respect to the size, they are very small. And so what
compared
happens
to control?
Yeah, compared to the wild type. Yes. What
Okay.
happens in plants is that generally when you have a circular structure, you have a lot of pressure inside and that cannot withstand. So what do plants do is they try to make a puzzle shaped cells because the pressure can be dissipated. So what happens in the mutant is they don't effectively make the puzzle shaped cell. They are still withstanding that whole pressure inside. So we are trying to investigate how are the mechanical properties being changed over there.
Okay, good, nice, good luck.
Thank you, Vishwadeep. You really got key suggestions from our expert mentors. We will move to our next speaker, which is Olivia. I can see you, Olivia. Could you please confirm?
Am I audible?
Yeah, you are audible and your video is
I'll share
also
the screen. Yeah.
Okay.
Are my screens visible?
Yeah, it is.
Is it in full screen? Just a second. Is it in full screen mode?
Not yet. Okay, it's now. Go ahead.
Now?
Yeah, go ahead.
Okay. First of all, I would like to thank India Bioscience for giving me this opportunity to present my work. Hello, everyone. I am Olivia Majhi, doing my PhD from Department of Zoology, Banaras Hindu University, under the supervision of Dr. Devanjan Sinha. And the topic of my research is A Tale of Two Stem Cell Lineages, Unraveling the Role of Superoxide Dismutase 1 as a Novel Regulator. The regulation of multi-stem cell lineage is a highly coordinated process influenced by several intrinsic and extrinsic factors. In order to study these multi-stem cell lineages, we have chosen Drosophila testicular stem cell niche for our study. So in this posterior portion of this male Drosophila lies the pair of testes. So this pair of testes is a coiled tubular structure, and if we zoom into the tip of this coiled structure lies the testicular stem cell niche. Inside this niche are the hub cells. So these hub cells are basically 10 to 15 group of cells which are terminally differentiated somatic cells. Surrounding the hub are the germline stem cells. So here, one germline stem cell is surrounded by two cyst stem cells. So, and they form a rosette-like pattern surrounding the hub. When these stem cells asymmetrically divide, the cells that are present near the hub, they retain the stemness, and the cells that are present away from the hub, they differentiate. So this germline stem cell differentiated from the gonial blast, and the cyst stem cells, they differentiate to form the cyst cells. Here, the gonial blast again goes for 4 rounds of transit amplification, and they form 2-cell, 4-cell, 8-cell, and 16 cells, and ultimately differentiate to form these spermatids. But whereas these cyst cells, they don't divide as such, but they elongate and they accommodate these dividing spermatogonia. Now, this germline stem cell lineage is consisting of these germline stem cells, and the somatic lineage is comprises of the cyst stem cells and the hub. So this stem cell basically reside in a specialized microenvironment known as niche, which kind of determines their self-renewal, proliferation, and differentiation process. There are several factors that determines this kind of behavior, and ROS being one of them. ROS, that is reactive oxygen species. We all know that higher amount of ROS is detrimental for the cell, but basal level is actually necessary for their proper functioning. And recent studies have showed that ROS has evolved as key molecules influencing cell-pet decisions like maintenance and differentiation. We know that these ROS redox-dependent events are basically cell-intrinsic in nature, meaning the cell which is actually producing the ROS, they are being affected. But our observation suggested that the cells that are actually producing the ROS is affecting the neighboring cells and kind of establishing ROS as an intercellular communicator. And thus, we want to broadly look into how this ROS actually function in the maintenance of this testicular stem cell niche. In order to perturb ROS in this system, Drosophila has a very interesting system where we can do that. So that is known as GAL4-US system. In this system, we can upregulate or downregulate a gene of interest in a tissue-specific or cell-specific manner. So there are two transgenic lines. One is known as GAL4 driver line, and another that is US responder line. US, that is upstream activating sequence. Now, when these two transgenic lines are crossed, then the GAL4 from one line that sits on the US and leads to the transcriptional activation of a gene of interest. Now, inside the cell, in order to maintain the redox homeostasis, there is a balance of antioxidant and oxidant. To increase the ROS, we have downregulated several antioxidant enzymes. And when we have downregulated SOD1 and SOD2 in our background, we have seen some interesting phenomenon, and that we have carried on further. So we have set up this crosses at 29 degree because this GAL4 is an yeast transcription factor which works best at 29 degrees Celsius. And then we have dissected the 3 to 5 days old testis. And then we have performed immunostaining, and followed by we have scanned these on confocal microscopy. Now, so we have increased, first of all, we have increased ROS in the somatic lineages using the somatic lineage specific GAL4, that is TJ GAL4, which thrives in the hub and CYSC. And then what we have observed that there is an increase in the number of TJ positive cells. TJ basically marks the early cyst stem cells and the differentiating cyst cells. Simultaneously, an interesting observation we found out when we have increased ROS in this kind of somatic lineages, the neighboring germline stem cells, their number reduced. So here, the germline stem cells, we have counted those cells that are attached to the hub. And here we have observed when we have increased ROS in the somatic lineages, there is a decrease in the number of germline stem cells with the overall reduction in the level of VASA. So here VASA is the marker for germline stem cells. Now we have also downregulated SOD2 in this background, and we have observed that there is an increase in the number of TJ positive cells and reduction in the VASA positive cells, but the intent was less compared to the SOD1, and hence we have further carried on SOD1 in our future experiments. Now we wanted to check like whether this proliferation, like what is the state of the rate of cellular proliferation in this background. And we have used a sensor line for that, that is known as FUKI. So FUKI stands for fluorescent ubiquitin cell cycle indicator for tracking the cell cycle stages in Drosophila tissues. So here you can see these green cells are the cells that is present in the G1 phase. The cells that are present in the S phase are demarcated by red. And the cells that are in yellow, that these are at G2 and M phase. And here we observe that when we increased ROS in these somatic lineages, that there is an increase in the number of cells in each of these phases of the life cell cycle. But there is no as such change in the percent of cells, denoting that there is an increase in the rate of cell division, but as such, the cells are not present in a particular phases of that cell cycle. Then we have counterstent with another marker that is known as ZFH1. So this ZFH1 basically marks the early cyst stem cells. And here we see that these cells, when we have counterstent in this background, that these cells are significantly higher in case of G1S, G2M phase. Then we checked for another mitotic marker, that is phosphohistone 3, and we also observed that there is an increase in the level of PH3 when we have increased ROS in the somatic lineages. Now we wanted to check like what is the differentiation status, like whether the cells are only dividing or differentiating. For that, we have used another marker that is known as EYA. So this EYA basically marks the differentiating cyst cells. And here we observe that there is an increase in the level of cyst stem cells as well. But with that, we have observed a very interesting thing, that there are a few cells which is expressing both ZFH1 as well as EYA, and their numbers are significantly higher when we have increased ROS in the somatic lineage, suggesting that this early expression of EYA, like the differentiation marker, resulted in a population of differentiating cyst cells which also express the stem cell marker, that is ZFH1. Now, simultaneously, we checked what is the status of the neighboring germline stem cells. There also we observe that the differentiation status of those germline stem cells there is early differentiation of this germline stem cells. So for that, what we have used, so here we have used another marker, that is BAM, known as bag of marbles. They kind of mark the 2, 4, 8, 16 cell stage of the gonial blast. And here we can see that in when we have increased ROS in the somatic lineages, that these cells lie close to the hub, suggesting early differentiation of these germline stem cells. For that, we also checked for another marker, that is alpha spectrin. So the alpha spectrin, so in the germline stem cells, the cells that are present nearer to the hub, they have round-shaped fusome, and as they divide, they have branch fusome. And when we have calculated the distance of this branch fusion from the hub, we observe that in high ROS background in somatic lineages, this branch fusome is lying nearer to the hub, suggesting early differentiation of these GSCs. With that, we want to check, like, are these germline stem cells are actually adhered to the hub, or whether they're receiving the signal properly from the hub, or like what is the status of that scenario. So there we check for E-cadherin level, whether these germline stem cells are actually adhered to the hub properly or not. And there we found that in that high ROS background, the level of E-cadherin is significantly lower, suggesting that these cells are not getting proper signal from the hub, and ultimately they are differentiating. So in order to summarize like what happened when we have increased ROS in the somatic lineage, there we observe that there is an increase in the number of cyst stem cells and cyst cells with the decrease in the number of germline stem cells. And both of these cell types are actually early differentiating. Now, we kind of thought of like here, first of all, we have increased ROS in somatic lineages. Now comes the question, what happens if we increase ROS in the germline stem cells? Will we observe the same kind of phenomenon, like whether the ROS will affect the neighboring somatic stem cells or not? Let's see what happens. So when we have increased ROS in the germline stem cells using nanos Galfor, there we observe that there is a decrease in the number of GSC cells. That has already been reported that when we increase ROS in germline stem cells, there is a decrease in the number of cells, germline stem cells. But when we checked the status of the neighboring cyst cells, we observe that there is non-significant changes in the level of TJ positive cells. Then we also checked into the differentiation status of these somatic stem cells, and there we observed that the level is not so significant, suggesting that when we are increasing ROS in the germline stem cells, they are actually affecting only the germline stem cells, not the surrounding somatic stem cell lineages as such. Further, we validated whether there is increased proliferation. So we checked for the Fuki, and also there is no such significant changes in the level of the cells that are present in different phases of the cell cycle. And then we observed that this increased ROS in germline lineage decreases the GSC number with negligible changes in the neighboring somatic lineages. And this also kind of indicated that there is a non-autonomous redox transmission that is happening from the cyst stem cells, and that is going towards the germline stem cells, but not actually happening in a reverse way. Now, what could be the possible reason for this difference in the behavior of these two stem cell types? We all know that mitochondria is a major source of ROS production. So now we wanted to check what is the status of mitochondria in these two cell types. And there we observe that the mitochondria present in the germline stem cells marked the vasa, they are fragmented, compared to the mitochondria that is present in the cyst stem cells marked by TJ, and which kind of suggested that these difference in their distribution of mitochondria could be a possible reason that they are behaving differently. And then we checked what is the ROS profile of these two stem cell types. There we observe that when we have checked the ROS using a GSTD reporter line, and then we see this ROS is actually peaking at the boundary of these two cell types. So basically, we have calculated the ROS present in the boundary surrounding the cells that surrounding the hub cells. And there we found that there's an increase in the ROS level that is peaking at the boundary of these two cell types. Now, when we have increased ROS in the somatic lineages, we observe that obviously there is an increase in the ROS level since we are down-regulating SOD1. But this peaking is actually shifting. That means the ROS that is producing from the cyst stem cells, they are kind of transmitting to the neighboring germline stem cells. And this is indicated by this overlap here. And probably this could be the reason that we are seeing the changes in the status of the germline stem cells number as well as their differentiation. Now, like, how is this elevated ROS in the somatic lineages is actually affecting this niche maintenance signaling and promoting their proliferation and differentiation? What could be the possible mechanism behind this? Now, when we are increasing ROS in this somatic lineages, we already know that there are three major signaling cascades that is involved in the cyst stem cell maintenance as well as their differentiation. So we checked their status, what is happening when we are increasing ROS. For EGFR, when we have stained with phospho-ERK, so activated ERK, and there we found that the level is significantly lower in comparison to control, which is not the usual way, because we all know that EGFR is ROS modulated. But in our case, we have not observed that there is an increase in EGFR. Rather, we have observed that there is a decrease in the level of activated ERK. Now we wanted to check, like, what is the status of PI3K-TOS signaling, because this signaling is involved in the differentiation of the cyst stem cells. Here we observed indeed that there is an increase in the level of the PI3K-TOS signaling. For that, we have used the reporter line for 4-EBP. And here we observe that there is an increase in the level of the 4-EBP when we have increased ROS in the somatic lineages. Then we checked for another signaling, hedgehog signaling, which is known to one of the major signaling cascade that is responsible for the maintenance of the cyst stem cells. And there we found, when we checked for the components of hedgehog signaling, the numbers of the hedgehog signaling, the components of these hedgehog signaling. So PTC and CI, this is significantly increased when we have increased ROS. And we have done the RT-PCR of the components of this pathway, and they are also significantly higher, suggesting that this pathway is actually ROS modulated. And in order to summarize what is happening in the signaling cascade, so when we are increased ROS in these cyst stem cells, the EGFR signaling is downregulated, and PI3K-TOR and hedgehog signaling are upregulated. Now we wanted to check like what will happen if we downregulate like PI3K-TOR and hedgehog signaling in the background of high ROS. Will this phenotype rescue? And yes, when we have downregulated TOR and hedgehog in the background of SOD1 RNAi, we observe that there is a rescue in the number of EJ positive cells, as well as there is a rescue in the number of VASA positive cells. So in order to summarize what we have seen now, so when we have increased ROS in the somatic lineages, what is happening? So this increased ROS is basically leading to the increase in the number of cyst stem cells, followed by the cyst cells, and decrease in the number of germline stem cells. And these two cells are early differentiated. Probably these are happening because of the different niche maintenance signaling, where we have seen upregulation of the hedgehog signaling, upregulation of the PI3K-TOR signaling, and downregulation of the EGFR signaling. Now, when we have increased ROS in the germline stem cells, there we observe that these increased ROS in the germline stem cells have actually reduced the number of germline stem cells, which is already known, but with a negligible change in the neighboring cyst stem cells and the cyst cells and the overall niche architecture, suggesting that here we have established ROS as an intercellular communicator, which is mainly originating from the somatic progenitor cells. And these cyst stem cells basically functioning as a source of both of their maintenance, and non-autonomously they are maintaining their neighboring germline stem cells. And here we have observed that hedgehog pathway is also being ROS modulated. And this SOD1, we have established this SOD1 as not only as a control for ROS level, but also as a novel regulator for maintaining the stem cell architecture and homeostasis. This work has been acknowledged by India Bioscience in their Right-on competition. And this work would not have been possible without our team at the cytogenetics unit where I work, and the funding agencies. And this work is in eLife as a reviewed preprint. And I'm happy to take questions. Thank you.
Thank you, Olivia, for the wonderful talk. We can move to our expert mentors for your suggestions and feedback.
Okay, I'll
Yes.
start.
Sure. Yes.
Hi, Olivia.
Yes.
Very
Yes,
nice talk. Very beautiful images.
Thank you so much.
Yeah. So I was wondering, you indicated that there was a sort of ROS generation by one set of cells, which was diffusing across to another set of cells, but it didn't happen in the opposite direction. Even when you drove the knockdown of the SOD in
Yes.
those cells, you still didn't have that. So what do you
Yes.
think explains that?
So when we have done the GSTD staining, that's what I was coming, like what could be the reason for this difference in the behavior of these two particular stem cells? So we know that for the ROS generation, mitochondria plays a very crucial part. So there, when we have stained with, we have checked the status of mitochondria, there we found that these mitochondria are more clustered in the somatic lineages compared to the germline lineages. So in the germline lineages, so they are fragmented. And when we have checked the redox profile of those particular, that is in control. So we have done this in control, like what could be the possible way that this is happening. So when we have checked in the control, like what is the status of this ROS profile of these two stem cell lineages, there also we found that normally the CYSC, the cyst stem cells are actually having more ROS compared to the neighboring germline stem cells. So this could be a possible reason that we are seeing a change in the behavior of these two kind of cells.
So I'm a little confused. How are you? You is important downstream, but what is the actual mediation of the ROS signal? How do you think the ROS, the change in ROS levels is actually being experienced by the stem cells in that particular niche?
So basically, ma'am, these hub cells act as an organizer. So when we are changing the redox profile of these cells, so they give signals to the neighboring cells, and like whether they will remain as stem cells or they will differentiate. So when we are changing the redox profile of this particular cells that is surrounding the hub, and these stem cells are actually, this ROS change in the ROS profile is actually triggering different niche maintenance signaling.
So I'm just wondering about what the mechanism of that might be.
Ma'am, yeah, that is, we are also finding out, like there we have not gone deeper into how could be these ROS. Yes, exactly.
Yeah. What approach would you take if you had, what would be the ideal way of being able to figure out what the mechanism is? Do you have ideas about that?
Like you're asking like how this ROS is actually triggering this niche maintenance signaling?
Yeah.
So already, ma'am, like we know, like EGFR, PI3K, TAW, these are redox modulated. We know that. So these higher ROS actually, they kind of changes the level of EGFR and PI3K TAW. So possibly this high ROS background is actually affecting this kind of signaling because they are already targets of ROS and they are triggering this cascades. And finally, we are seeing increased in the number of these kind of stem cells in our background. But we have actually
other ways of modulating ROS levels, which don't involve SOD itself, but other ways of modulating the levels of ROS, within the niche. Do you predict that you would have similar changes in fate?
So, ma'am, basically we have tried once, like we have tried paraquat feeding, like increasing ROS in the background, like what will happen. But the problem with that is like when we are feeding the flies with paraquat, so it is the whole cellular, like the whole organism is high ROS. So that is the main problem.
Yeah, an inhibitor, mediated thing like that would be too generalized, I think. I was just thinking of other ways.
Like for that, we use the Galfo system. Sorry, ma'am.
Yeah. Some other mechanism using, I don't know what the genes might be, but I was just wondering if there are other genes that would lead to alternate or, for example, increased or decreased ROS signaling in the opposite direction from what you have already done.
I will try, ma'am. Actually, we have looked for different antioxidant enzymes. So we have gone for catalase, PRX, but there we have not seen much changes in the level of these cells. So that's why when we have downregulated SOD1 in our background as well as SOD2, there we have seen a striking difference. But the difference was not evident when we have downregulated like catalase or PRX in our background. So I would look into like ways like how we can also increase ROS. But for now, the transgenic lines we have with us are these kind of lines available. Sure. So we have gone further with those studies.
Thank you.
Yeah. May I just ask a question as an extension of what Josna was asking? So you know that some genes need redox enzymes for activating their transcription factors. So maybe that will give you an idea as to how the redox reactive oxygen species are actually read. That is very important. And for example, AP1 is one enzyme in mammals, which has both DNA repair activity as well as redox activity. But this gene is not present in Drosophila.
Drosophila, right.
So there will be some other enzyme which, activates transcription factors like AP1 and so. So they need to be activated if your ROS is going to be effective in activating specific genes. Maybe you would like to look at some of them in the database to begin with. Yeah?
That's a great suggestion. I will look into the yeah.
May I ask?
Yeah,
Sure,
please.
sir.
Yeah. So, do you think this SOD1, the superoxide dismutase, it has some non-enzymatic role, something else that can be also contributing to the gene regulation, and that's why you see different effects in different cell types?
So non-enzymatic role, as in like I'm not
other
getting.
than other than redox related role.
Okay, so
Is
are
it
you're
possible?
saying like whether SOD1 has any other role in the development of these kind of so actually the sort we all know that these enzymes are mainly linked with antioxidant system. So here also we are trying to establish this as a regulator for the stem cell homeostasis maintenance. So it's not like they're only maintaining the ROS, but possibly if we just perturb the level of these antioxidant enzymes, we change in the niche architecture.
So the way to deal with antioxidant, like or oxidative stress in different cell types, embryonic versus your cyst stem cell, are those same or different?
Sorry, sir. Again.
The mechanisms to deal with the oxidative stress in your germ stem cells and the cyst stem cells, are those same? Do you know?
No, sir,
do you know
they're
anything
different.
about it?
They are different. That's why we are seeing a change when we are perturbing ROS in these two stem cell types.
So how
they
they are
don't.
different?
So basically, when we are increasing ROS in the germline stem cells, so you are seeing the changes like that is already well known.
You're talking about a phenotype, right? So I'm talking
Yes.
at a mechanistic level. How is it different? Same genes, same
So,
pathways are involved.
So possibly from our observation, I can say that these cyst cells are actually they have high threshold for the ROS compared to the neighboring germline stem cells. So their because they can resist the change, like when the ROS is increased, so they are not reacting to it. But when we are changing the ROS in the neighboring germline stem cells, they are kind of more affected by that.
So based on phenotype, you're hypothesizing, right? Based on phenotype,
yes,
you're hypothesizing that there are different mechanisms, right? But
yes.
nothing is known in literature, neither you have tested anything like that, right?
So I have not gone for the mechanistic approach, yes.
Achha. Okay. So
thank you, sir.
No, it's fine. Just one more follow-up question. So you could see when you decrease the ROS in germ stem cells, you see the decrease in the germ cell number, right? So what is happening to those cells?
Yes.
What is actually happening to those cells?
So these cells are actually differentiating.
So are they giving rise to cyst stem cells?
No. So germline stem cells, when they differentiate, so they will differentiate to gonial blast.
Only?
Yes. Yes.
That is for sure. There is no differentiation to, let's say, cyst stem cells or any other.
No, sir, that we have not observed. That's what we were thinking. Like when we have done this in the opposite way, we have changed. We have seen change in the differentiation status of the neighboring germline stem cells. But when we have looked into the status of differentiation aspect of these cyst cells, they are not being differentiated.
Okay. So just one more follow-up question. In the cyst stem cell, you could see that there's downregulation of EGFR, upregulation of HOG, and what is this? PISK-TOR, right?
Upregulation
Right, TOR.
Yes.
So, when you increase the ROS.
Yes.
In the same scenario, if you downregulate the HOG, in the same experimental setup, if downregulate HOG, what do you expect?
So that's I have already shown there. So just yeah. So here we have like a downregulated HOG in the background of increased ROS, and there we have observed
this is
that there
done
is a
in
decrease.
Cyst stem cells, right?
Yes, sir. Yes, sir.
Okay, I missed that. Huh? So, but
Yes,
what
sir.
do you see?
So, sir, here we have observed that when we have decreased HOG in the background of high ROS in this cyst stem cells, so there is a decrease in the number of TJ positive cells, that is the cyst stem cells, and also the germline stem cells, because they are also kind of recovered from that. So we got a rescued phenotype here.
Germinal cells are rescued. Okay.
Yes.
Fine.
Yes, sir.
Thanks. Good luck.
Thank you, sir.
Yeah. Dr. Joshna wants to kind of follow up on that.
Yes, ma'am.
I just had a quick question, which was to do with your detection of the actual ROS levels in C2. Remind me how you did it. You had some kind of a fluorescent
GSTD,
tag.
ma'am, yes, we have used GSTD marker, yes, reporter line.
So what, can you just explain to me how that works?
Yes, ma'am, this is actually a reporter line. So it's a sensor line. So when there is high ROS, so you will have more GFP. So because the GFP is tagged with the GSTD reporter. So in the so here we have calculated the GFP, and there we have seen that these when we have increased ROS, obviously that there is an increase in the level of GFP because the GFP is actually tagged with the GSTD.
So
and ma'am,
can you
also
just explain? I'm not aware of this this system. So can you just explain to me how that works? How does the ROS
Sure,
actually increase the fluorescence?
So ma'am, here the GSTD gene, the GFP is actually, yes. So the GFP is actually present downstream of this GSTD. So when the GSTD is active, so there you have the higher amount of GFP. So here when you're increasing ROS
GSP is what? GSTD is a ROS sensitive.
Yes, that is
factor?
glutathione transferase, yes.
Okay, but again, this is a reporter for something that's happening within the cell, but you're not actually seeing the amount of ROS itself. It's a sensor for the amount of ROS, right? So I was wondering if there are not direct sensors that are fluorogenic sensors for ROS itself, the actual change in the ROS levels.
Ma'am, we tried that. So we have done DHE staining, but the DHE staining as such, it's like I can't demarcate the cells there. So we have observed that there's an increase in the, like when we have compared the two levels of the DHE staining. So obviously there was increase, but there we were unable to mark those cells. And also we have done for mitosocs. So there we have the readout, but those were actually not a good marker to indicate the cells. So we have tried different ways, and actually visualizing ROS is a very tricky thing. Once we have started, like it just quenches very quickly. So that's the problem we are facing. Yeah.
Okay. That would be nice, because that would then give you, if you could crack that, then you would have a direct readout of in a spatially resolved fashion. You would be able to see the phase changes as well as the actual ROS levels, which would be very nice.
We tried for that. This was the best one we could found in that background.
Okay. Thank you.
Yeah. Thank you.
I'll ask a short question. I was kind of surprised that cell cycle kind of sped up. That you didn't see a G2 to M block, which I would have expected. Did you look at G2 to M checkpoints in the?
No. So we have not gone for we have not checked the checkpoints, like what is the status of the levels there. But when we have seen the cell like proliferation rates, so
Yeah, I remember the Fuji experiments.
Yes, sir. So there we have seen that there is an increase in the number of cells in every stages of this cell cycle. So there was increase. Yes.
Yeah.
So, but there we have not gone into, like, since there was increase in all the phases, and that's why there was no significant changes in the like when we have done percent of cells in the each phases of the cell cycle, there was no significant changes, because it's just the cell numbers are increasing in every phases of the cell cycle. So the rate of cell proliferation is like higher.
Can you measure the rate? Are you able to measure the rate? Can you measure the rate of proliferation?
So I have not done it. I have not done it.
Yeah,
I've just checked how many cells are present in the particular phases of the cell cycle, but like I have not gone for the checkpoints and like how many cells are going to that cycle. That I have not. That would be.
I mean, the checkpoints would be good, but you can measure the actual rate of cell cycle. Okay, using some for example, a BRDU EDU kind of tandem.
EDU, yes, incorporation, yes, sir.
Yeah.
Yes, sir. That yes.
Follow an EDU pulse with BRDU, you'll be able to measure the proliferation. Okay, and whether the proliferation is happening now or earlier, things like that. Okay.
Yes, sir. Yes, sir. That thank you for the suggestion. I'll check that. Yeah.
Okay. I'm done.
Thank you, sir.
Thank you, Olivia.
Thank you so yeah, absolutely. Go ahead.
Thank you.
Rizija.
Thank you.
Yeah,
Okay.
thank you to our expert mentor. And Rizija, just before you close, thank you to our PhD speaker, thank you to your audience. And this is the giveaway, our latest e-compendium on challenges and solutions to better support PhD experiences in the life sciences in India. And that's led by my colleague Ankita Rathore, who's Program Manager Science Communication at India Bioscience. So there's lots always going on at India Bioscience, just in case you haven't figured. But this is the giveaway, and the winner is Dibyo Mazumdar, who interestingly has already sent us an email to claim his giveaway. So well done, Dibyo. Well done. Well done. And you are our 10th winner of the giveaway. So thank you so much, Jyotsna, Raj, Prasad, Surendra. Thank you so much. Took up a lot of your time. Hope you enjoyed it. Yeah?
Yes,
Great.
yes, yes, yes.
Great.
Yeah, of course, yes.
Very nice.
Yeah, well done, everyone.
So
Thank you so much.
nice
Thank
to see
you.
very confident
All the best.
presentations.
Yes.
Yeah.
And good luck to everybody.
So much to learn for me also. So great.
Yes.
Thank you so much. Rizija, you can just say thanks, son. We can call it a day. Yeah.
Okay, thank you everyone. This was our last session of PhD Cafe. Thanks to all the PhD speakers for taking us through the fascinating world of developmental biology. And thank you so much to our expert mentors for their priceless suggestions and feedbacks. So no session tomorrow. I'm definitely going to miss joining at 10 a.m. to 5:30 p.m. So it was really amazing thing. And a big
Yeah.
thanks to India Bioscience for giving me this opportunity to be a part of planning, outreach, and moderating the PhD Cafe. It surely has been a journey. Thank
Certainly
you.
has been.
Thank you.
Certainly. And I think we all at India Bioscience are going to need 2 days of digital detox after 5 days of PhD Cafe. All right. Thank you so
Definitely.
much, all of you. Have a good
Bye all.
weekend.